Someone, you want me to start?
So we are on live now.
It will take, I think, 2-3 minutes to complete the na.
66, 69, 74, 75, 79. Yeah. Oh, 92. Great. 100 हो गया. चलो करो ना अभी.
करो ना.
करो ना मतलब not that करो ना. On some group, I was told like 90 days तक वायरस रहता है. मैं बोला कि यार इनको पता ही नहीं कि day 3 करना है कि day 5 करना है. इनको ऐसे ही करता रहेगा. 90 days है कि 24 days है. Hi Akash.
Hello, mute पे था, sorry. Hello, sir.
Who else is remaining?
I think.
Yoshita,
Yoshita is there, I think.
because
हेलो सर.
hello.
तुम्हारा साउंड बहुत कम आ रहा है योशिता. थोड़ा सा.
हेलो सर.
Hello.
ठीक है.
हाँ ठीक है. चमन जी, हम मेरे को लगता है हम स्टार्ट करें. आप इंट्रोडक्शन स्टार्ट करो तो फिर हम आगे बढ़ते हैं मेरे ख्याल से. अब है ना.
Yes.
क्योंकि ऑलरेडी 49 हो गए, फिर हमारे पास टाइम नहीं रहेगा. स्टार्ट एग्जैक्ट 3 o'clock हम चाहते हैं कि हो जाए प्रोग्राम स्टार्ट.
CP sir.
Sir. There is a problem with my image, I think.
Yes.
You have to remove your background, I guess.
No, that already removed. I think how come it has come appeared again?
No, you go to the settings.
It's invisible man, eh?
Yeah, it's looking like Mr. India coming here.
Hollow man.
I want to know how to create that background. I want to learn that. I don't know.
There I also want to learn this thing.
I saw first with Alex. Then, Alex, and then Vijay. I thought every time it's the same or something different, and it's looking totally. Where is it sitting?
Alex sir तो गैलेक्सी में बैठे रहते हैं.
You can take lessons from me.
Very good afternoon and welcome to this show, India's first online international symposium, expert meeting for the best practice in clinical embryology. I think this platform is created by Dr. Ved. Thanks for Dr. Ved for creating such a good platform to share the knowledge. I would also like to introduce the embART-2020 Pre-Congress Masterclass Coordination Team. And this team is consisted of Dr. Rahul Sen, the Chief Embryologist at Vasundhara Hospital, Jodhpur and Jaipur, Dr. Sanket Dhumal, the Chief Embryologist at Seema IVF Fertility, then Dr. Paresh Makwana, the Chief Embryologist at Wings Hospital, Ahmedabad, and Dr. Akash Agarwal, the Scientific Director at Hegde Fertility, Hyderabad. I would like to hand over this platform to our coordination team. Over to the coordination team.
Hello, everyone. I would like to call on stage Miss Yoshita. She is a embryologist from Delhi area. She'll be taking on the session next forward and introduce our founders and co-founders. Yoshita, it's over to you.
Thank you, Akash, for your kind intro. I would like to introduce our founders and co-founders of iHear Academy. Firstly, I would like to introduce Dr. Ved Prakash. He's the Lab Director of Southern Fertility and IVF, Delhi NCR. and second, Dr. Sanjay Shukla, sir, who is the lab director at Baheti Hospital, Jaipur, and Shivani IVF and Fertility Center, Jaipur. He has done his PhD in life science, and Dr. Charudha Joshi, who is a medical director at Gene India ART Bank. So over to you, sir.
Trisha ke dekhe na.
Thank you, Yoshita, for the introduction. And respected dignitaries, my dear friends and colleagues, on behalf of IHRA, I welcome all of you. And of course, for supporting drive-by system. And IHRA is not a non-profit online platform for knowledge sharing and experience aiming at quality information in the field of infertility, a field which is evolving by leaps and bounds. Newer treatment protocols and robust technology are emerging rapidly. It is very easy to get carried away by these enticing development like embryo genomic and time-lapse monitoring. It can lead us to forget the importance of our basics like dish making, blastocyst culture, vitrification, which are among the foundation for our day-to-day success. Hence, this pre-symposia masterclass was designed to strengthen our basics, our day-to-day embryology. So I welcome you all to embART-2020, and I invite Dr. Shukla to proceed, please.
Thank you, Dr. Ved Prakash. As you mentioned, this online education platform was conceived with the idea that probably we can utilize the lockdown period when everyone is at home, and that's how we started working on this. Of course, the idea of having our own online platform to disseminate knowledge, the scientific advances that are coming up in the field of embryology and ART was there for a long time, but we could materialize it with the help of our young team. And I mean, it's heart touching and heartwarming the way they did the whole work day in, day out, and without tiring. I mean, of course, I was not able to attend all the meetings and interact with them, but what I could get from all the other members of the team, that even their meeting lasted for, say, up to 2 a.m. in the morning. So it was a never tiring job, and they did it relentlessly. And I'm sure the next day also, all the participants would witness a very sound and very meticulously designed scientific feast. So I welcome all on behalf of IHRA and embART. So over to you, Yoshita.
I think Dr. Charu Joshi is not available.
Yeah,
so,
probably he is in the transit, so he may not be able to join us.
So maybe Akash will take care of this thing. Akash, are you there? Akash?
Okay, so we'll take our session forward. We'll be going on to the first topic, which is tumors in vitrification. The speaker is Lodovico Parmigiani.
Akash, sorry to interrupt you, because we have actually 3 minutes to go. So I think we have to wait for things.
Okay, sorry.
It will start around 3 o'clock.
Yes.
So we can start at 3 o'clock.
3 o'clock. Fine. I'll stop sharing.
Yeah. No, you can share. There is no problem, but start exact 3.
Okay, fine. I would say they were very kind words from Shukla sir for all of us. And we take the opportunity to thank him so much. We always require your guidance, always of the pioneers and experts in the field, and always the seniors. So this was designed keeping exactly the same thing in mind, that we might go to the highest level, like Ved sir was saying, that we might go to the highest level, but we might sometimes forget our basics. So these are the basics which will help us in our day-to-day practice. And if we go wrong anywhere in the basics, then our entire culture system and our entire results will go wrong. So I just hope, I'm waiting for our program to succeed, and I hope that all the participants can learn something and will be able to utilize in a much bigger way. And I welcome them all even for the tomorrow's event, which is going to be on a larger time scale and equally good quality.
I just wanted to say that when the lectures are going on, maybe we need to mute everyone, because there's a lot of background noise coming. So someone will have to take care of that. Even when Sanjay sir was saying, there was a lot of disturbance behind, so you can't hear the speaker properly.
Yeah. I think Mr. Chaman, are you there? Mr.
Yes,
Chaman?
sir. Yes, sir.
So you have to mute all the except speakers. You please mute all of the participants and everyone on the
Yeah, sure.
Hello, Sujata ma'am. Hello, Sapna.
I would request all the participants to be on time, because Dr. Parmigiani has to go. He has some other commitments.
Thank you.
I would even like to specially thank our international faculty for taking out their precious time and agreeing to present for us and for Indian participants, all of them, and abroad as well. So shall I start, sir?
Yeah. Please go ahead.
So we'll begin our session today, and the first topic would be QMS in vitrification. It is being taken by Dr. Lodovico Parmigiani and being moderated by Dr. Parshuram Gopinath. Dr. Parshuram is a clinician as well as an embryologist. He's a medical director for CMAR Cochin and the scientific director as well, where he's having the teaching faculty at Apple Hospital. He's no novice to awards, and he has multiple awards to his credit, and he's a member of reputed fertility committees, and is on the managing committee of ISAR and ISAR accreditation committee and executive committee member of ACE India. Now I hand over the stage to Dr. Parshuram.
Thank you so much.
Dr. Parshuram, sir.
Am I audible?
Yes, sir.
Thank you so much. And it's indeed a pleasure to be a part of this, and sincerely congratulate Vedji, Sanjay, Charuji for this amazing initiative, and the whole young team of Akash, Sanket, Rahul, the whole young team who's kind of nicely bringing this program to a good level. And to begin with, my job for the day is to introduce our first speaker for this conference. It's truly an honor to introduce Dr. Lodovico Parmigiani. He's the head of embryology of the Next Clinics International. He's an IVF lab director at the GynePro Medical Center in Bologna, Italy, and advisor for biotech companies. He's invented and developed and optimized devices and methods for clinical embryology. He's a key opinion leader in ART and also the founder and CEO of Netlizer. It's an innovative startup company which talks about sterilizing liquid nitrogen. That's what I kind of found out. So looking forward to hearing from you, sir. It'll be a pleasure. Over to you, sir.
Okay, can you hear me? I start to show my screen. Yeah.
You can start sharing.
Okay. Just confirm that you can see my screen. Can you see my screen? Okay. So let's start. Thank you. Thank you very much for your kind invitation. It's an honor and also a pleasure to talk about quality management system in vitrification. I love this topic. So just confirm that you see the slide going ahead, because sometimes there are blocks. But so this is my affiliation as already described, and my disclosure. Okay. So when we consider human gamete and embryo cryopreservation, nowadays this is integral to assisted reproductive techniques because optimizes treatment management and results. We know that cryopreserved cells can be stored for years and also transported between authorized assisted reproductive centers. Of course, we need to put in place strategy and procedures to minimize physical shock and contamination. So when we are evaluating the introduction of a technique in our laboratory, or we are monitoring the performance of the technique that we are performing in our laboratory, we need key performance indicators. KPIs used in cryobiology, in gamete and embryo cryopreservation related to cryosurvival. These are typically assessed immediately post-thawing or warming. And we evaluate the return of apparently normal function or morphological development. So, all of us have the perception on how well reproductive cells and tissue should cryosurvive. But we know that KPIs require precise definition and objective standardized methods for their determination. Just to give you an example, in the case of oocytes, we need to observe the proportion of intact oocytes post cryopreservation, and then we can also check the developmental competence such as fertilization, cleavage, blastulation, until also the clinical outcome parameters such as pregnancy and implantation. So, if we consider oocyte cryopreservation, nowadays we know that it is possible to cryostore for long-term oocytes. We know that oocyte cryopreservation is a technique which is consistent, effective, safe. With vitrification, it is possible to obtain a very high survival rate, and the benchmark of a vitrification program on oocytes, it is around 90% of survival rate. We know that frozen oocytes, vitrified oocytes, perform as well as the fresh. So the same is for embryos. Sorry, I jumped directly to oocytes and embryos. I did not consider spermatozoa because we all know that they are much more resistant than oocytes and embryos. Okay, so also for embryo, we have the information that we can obtain long-term cryostorage. We know that this technique is consistent, is safe. The benchmark for a good program of embryo or blastocyst vitrification is 99%. So this KPI is the highest that we can measure in our IVF laboratory. There is no other KPI so close to 100%. Of course, we know that vitrified embryos perform as the fresh embryos because we are experiencing in our laboratories that the majority of embryos now are vitrified to be used for frozen embryo transfer. It is to overcome problem in the uterus and to adopt a freeze-all strategy. So I introduced vitrification. Just to recap what vitrification is, we know that intracellular ice crystals have a detrimental effect, and this was well known since 1940. So we need to vitrify the cell, and vitrification is the conversion of superviscous liquid into glassy state when cooled below its glass transition temperature. It is very important to keep in our mind that any material can vitrify, and this is directly dependent on viscosity and cooling rate, and then inversely dependent on the volume of the sample. That's the reason why we use very thin carriers, and we increase the viscosity of our cells by adding permeating cryoprotectant and by dehydrating these cells. What we do to have the maximum cooling rate, we plunge directly the cell in liquid nitrogen. So, when we perform this vitrification, especially if we use open carriers in which we put the embryos directly in a strip on the carrier and we plunge it directly in liquid nitrogen, this in order to have, as we said before, a very high cooling rate, which is a technique very efficient with oocytes, we have potentially the risk of contamination, culture medium at forming through the contact between the cells and the other surface of the open carrier, if the liquid nitrogen is, or the nitrogen vapor that we are using, is accidentally contaminated. So when we perform vitrification, so we can use different systems. We can use open system, as we introduced before, in which we have a thin strip in which we put the oocyte or the embryo directly in this strip that can be a polyethylene strip, and we plunge it directly in liquid nitrogen, then we close it. Or we can have a straw closed system in which we put our oocyte or embryo in an inner strip, in an inner straw, and then we enclose this straw in another straw that can be sealed, and then this is plunged in liquid nitrogen. Or we can also have different systems, such as a straw closed system, or a system that use nitrogen vapor for vitrification. Or we can also have an automatic closed vitrification system. So, which is the potential way of contamination of the samples and also of the environment. So, if we accidentally have liquid nitrogen which is contaminated, we will have contaminants frozen in liquid nitrogen. So these contaminants may adhere to the surface of the carrier and then stay in the surface of the carrier. This was what we, we thought in 2011, but we need to keep in mind that in this pandemic, we need to keep in mind that if we have the liquid nitrogen that is contaminated, also the vapor arising from this liquid nitrogen can be contaminated. And if we use 1 or 2 liters of liquid nitrogen in near to our worktops, we can contaminate all the worktops. Anyway, this is the hypothetical model of contamination of cells. So when we plunge the carriers in the culture medium at 37 degrees, the contaminants awake, and then we can have contamination. Of course, we pick up this cell, we move the oocyte of the embryo to the second well for diluting the cryoprotectant, and then again we move it in another well in order to perform washing, and in this way we dilute the potential contamination. So the risk of hypothetical contamination of the cell is very low. Anyway, as I described before, even when we use closed system, accidental contact with liquid nitrogen and nitrogen vapor may occur during storage and warming of material. The real risk, so it's not related with open or closed system, but with the liquid nitrogen and nitrogen vapor themselves. If we think that a box with 2 liters of liquid nitrogen that we put near our worktops is contaminated, just to give an example with COVID, this can contaminate worktops, operators, cells, and maybe at the very end, the patient. So the fact that infection traceable to cryo storage have not been reported after millions of embryo transfer may be due to reduced dosage of infective agent during laboratory procedures. However, the risk of infection is real, especially when new highly infective agents arise. So we use liquid nitrogen in healthcare, but this is used also for food and beverage. We know that liquid nitrogen can be easily contaminated because contaminants were already found in the last decade in cell cryobanks. Viruses were found, bacteria, fungi. So contamination creates risk for biological samples, patients at the very end, and also food quality if we use it for food. There are some applications that requires sterile liquid nitrogen. The liquid nitrogen is sterilized before use, and one is for sure human-assisted reproduction for vitrification, warming, and cryo storage. We are sensitive to this problem. During cryo storage, the potential sources of contamination are, of course, other cryopreserved samples stored in the same vessel. So we are very worried in this period that a patient can bring infected cells, but we are reassured. Yesterday, I noticed an article from a Spanish group telling that the oocytes were of COVID-positive patients were without the virus. So this is very reassuring. But the real problem is not the cross-contamination mediated by the cell. The real problem is the liquid nitrogen itself. We have no control about the liquid nitrogen from the manufacturing to shipping and to the final use in our laboratory. So we know that in the tank, the contaminants can be in the aggregated phase, ice and sediment, which entrap viruses, bacteria, and also fungi. So COVID-19 is an airborne virus, and this can come in contact with liquid nitrogen, nitrogen vapor in any moment. For example, we had information that last month, some chicken wings that were, of course, frozen, not with liquid nitrogen, but in the surface of chicken wings, there was COVID-19, and these chicken wings traveled frozen from Brazil to China. So we can understand that liquid nitrogen is the ambient in which we can vitrify the virus, and this can be conserved. Okay, can be stored for many, many years. So we have information about aerosol and surface stability of COVID-19, but no information about the presence of COVID in liquid nitrogen. This virus is a potential time bomb. So the use of contaminated liquid nitrogen or nitrogen vapor risks virus awakening and the contamination of human cells, environment, and operators. So nowadays we know that it's possible to easily sterilize the liquid nitrogen by using UV radiation. UV radiation to kill a virus, the UV radiation necessity is very low. For hepatitis, it's 8,000 microvolts second for centimeter square. For coronavirus, is 10,000. For example, from the last decade, I worked to develop this device that give more than 10 times the energy necessary to kill viruses. So if we consider that when we handle the cells, we do many handling and steps. Any of these steps, this is just, just to give an example, the shipment of oocytes from Spain to Italy, any of these steps can give us potential contamination, because we use liquid nitrogen and liquid, so in any of these steps contamination can occur. Also during shipment. So you can understand that the use of liquid nitrogen is not safe. So what we do, what we can do, we know that multiple washing is effective in decreasing infectivity and rendering oocytes or embryo free from viral and bacterial pathogens. So the suggestion is to, wash, also after warming, wash the cells, adding additional washes. But we can also eliminate the problem at source by using washing with sterile liquid nitrogen. This was demonstrated 6 years ago, or 8 years ago, sorry. So what we did in this study, we contaminated, we performed a forced contamination of more than 300 carriers, plunged they were immersed in contaminated liquid nitrogen. And then half of these were washed with sterile liquid nitrogen, and 100% of these carriers were decontaminated. No contamination was observed at warming. So the washing with sterile liquid nitrogen is the easiest, straightforwardness method that we can use in our laboratory to minimize the contamination coming from liquid nitrogen. This is also a reliable trace for quality assurance and any strict regulation. So, of course, then we should be able to demonstrate that we are cleaning our dewars, and of course we can use chemical cleanings or other methods. This method which use also UV radiation. Keeping in mind that after we have decontaminated a tank, immediately after we may also have another contamination. So the final washing, I think, is very important. Regarding other problems, when we handle stored specimens, please, we need to always to keep in mind that we need to wear appropriate protection for body, shoes and hands. We need air sensor, portable if it is not possible to have an air sensor inside a cryo tank. There are some precaution that we need always to observe. So we need to check in any moment the situation inside the cryo bank. We need to use appropriate signals. We need to have the oxygen mask, and we also need to have a controlled access and also an emergency system to open the door to put in say the operators. Again, I recently commented an article on human reproduction with which Chinese group was declaring to monitoring the liquid nitrogen level manually. I think that, of course, now we know that there are also systems with the artificial intelligence, but always, if we cannot recur to these very sophisticated methods, nowadays a probe inside in a tank is not so expensive. So we need to have a spy system to monitor the temperature inside the tank, and also we need to have a very clear system, signal system to fire police or other personnel that can help us in case of a disaster. of course, we also need to have a very well detailed disaster recovery plan. So I finished in conclusion. What I said is that human reproductive cell can be stored for years, transported between authorized centers. We know that frozen cells perform as well as the fresh counterparts. We need the KPIs for monitoring the performance of our cryopreservation program, our vitrification program. We need appropriate procedures to minimize physical shocks and contamination during handling, transportation, cryo storage. We need appropriate procedures to reduce operator and specimen injury during handling and cryo storage. And my plea is not to underestimate the potential risk of liquid nitrogen and nitrogen vapor mediated COVID-19 infection. Thank you. Thank you very much for your attention.
Thank you so much, Dr. Lodo. Do I give in the questions right away because he needs to leave, or should I wait for the session to be over?
We can take the questions, sir.
All right. It was a quite enlightening talk, Dr. Lodo, and it did kind of, again, the sterilization of liquid nitrogen does kind of come over a lot of debates and other questions asked about it. One very interesting question that I saw in this particular question and answer session is how do you actually check for contamination? How in this liquid nitrogen? How do we know if the liquid nitrogen is contaminated or not? And then how once you sterilize it, do you have kind of, when you say washing it, do you have any videos or you have some YouTube links you can share so that it'll be useful for all the people to how to kind of use a sterile liquid nitrogen to wash the liquid nitrogen and all those things?
Yeah, thank you. Yes, well, what we have to do, we have to consider liquid nitrogen always contaminated, unless we don't have a sterilizing system that feed our cryo tanks, which is impossible because we did already develop it with our company. But so we need to consider that liquid nitrogen can be contaminated in any moment. So anytime we open the tank, this can be contaminated. So what we can do is to to minimize this contamination. So to demonstrate that we are doing periodic cleanings, and also that anytime we pick up a specimen, we are washing this specimen. And then, yes, of course, I can share also videos in which you can see how easy to use and straightforward it is to use this box to sterilize liquid nitrogen. Nowadays we use boxes that are dirty. We can clean it, but at the very end are dirty. And this is the missing link of the chain. So imagine that you have a sterile box with sterile liquid nitrogen. That's it. This is what it was missing. And this is what I worked in the last 10 years.
Thank you. And so when it comes to the fact that we are sure that now the liquid nitrogen is going to be sterile, so do you at this point of time opt for a closed system or an open system of vitrification?
No, because at the very end, now this is what I realized. The real problem is not the open or closed system. So the contamination of the cell is negligible. The real risk is that when we bring, I got this, when we bring a box with liquid nitrogen in our worktops, we can contaminate the worktops. So the real problem is not in the oocyte. The real problem is in the liquid nitrogen that we use. So at the very end, using open or closed system, yes, of course, closed can be closed cryo storage. In my opinion, is better. But at the very end, if you have the possibility to wash a carrier before using, you are doing your best to minimize any contamination. The real problem is the liquid nitrogen. If you bring in your worktops a box, a dirty box with contaminated liquid nitrogen, you are contaminating your worktop. That's it.
Another question that I wanted to ask is how do you kind of sterilize the liquid nitrogen? When you say that you have a separate box with the sterile liquid nitrogen, where does that come from?
It's UV radiation. So it's very simple. The method is UV radiation. We know and we read in this period that UV is effective to kill COVID. So we give UV radiation to sterile trays inside a box which is sterilized with UV radiation. This is gamma sterilized by gamma radiation, and then you fill it with liquid nitrogen and sterilize. 10 minutes and that's it. 10 minutes, that's the time you need to write in the carrier.
How long do you need to keep it inside that to get sterilized? Like say, for example,
you need to bring it inside your workstation in which they are filtered. So you use your gloves, mask, and air filtered. Better if you put this inside your workstation, but you keep it near your workstation. Do the sterilization, take out, goes with this, and then you got it. You do the vitrification, and then you can use back this liquid nitrogen. You fill it another box, you do another sterilization, you can use it again. So in this way, you are not also using extra liquid nitrogen. You can always sterilize the same liquid nitrogen, refilling, and that's it. So it is once you start, it's impossible to go back. Of course, this is an extra cost, but it's a small cost to have high safety. So you are safe in front of the patient.
Yeah, but one last question from my side. So how do we actually know you sterilize it? How do you know that it is sterile?
Thank you. Yes, we have a label. So we give the proper irradiation when we finish the device. Give information about the irradiation. It was given, and then we also have a sensor that change color inside the box, and when the sensor change colors from yellow to green, we are given the right energy.
Thank you. So I think somebody did ask for the duration of the UV radiation exposure.
10 minutes in this system. Of course, what is important is that this cannot be universal. It is universal for this device. We calculated everything, and in 10 minutes, you have the sterilization. This can change depending on the humidity, level of liquid nitrogen, outside temperature, but can be from 10 to 11 minutes.
Thank you so much. And I think, do we have any more questions that I need to ask? I think we've almost...
And then at the end, also the sterilization are registered in a portal and then put in safe with blockchain. So, if you have any legal controversy, you can say, I did my best.
So I think we've done with almost all the questions. I think some people do have asked regarding how to procure your kit and what is the cost of it and all. So probably if you can share a link on something so that probably people can contact you directly and see how this can be taken, because I don't think a majority of the centers, and at least in our country, is using.
Yeah, this is a new product, and we are starting in Italy and in Europe. Many centers are starting to use, and of course, hopefully this will be distributors also by big brands.
Okay. So anyway, thank you so much, and if
welcome.
I may close this part of the talk and move on to the next session. Saruj, can you take over as the next chairperson?
Thank you. I would like to invite now Dr. Sanket. He'll carry forward the session along with Dr. Saroj Agarwal.
Thank you, Dr. Akash. It takes me immense pleasure to introduce our next moderator for the session, dear Dr. Saroj Agarwal. And she's a lively person and very enthusiastic person who actively involves in academic events. And currently she's working as a chief embryologist and scientific director at Care IVF Kolkata. And she's associated with Care IVF since 2007. And she was involved in successful setup of first PGS and PGT facility in West Bengal. And also she has, to her credit, she is also involved in USET freezing program, first of its kind in Eastern India. and she's executive committee member of Academy of Clinical Embryology India, and she's a member of IFS and ISAR, and she also hold a gold medalist during her MSc. Over to you, Saroj ma'am.
Thank you, Sanket. Thanks a lot. I thank iHera to give this opportunity and to bring this wonderful platform to all of us here. And we have the pleasure of having Dr. Manal El Sahidi here with us. Today, she is currently working as a scientist with Fujifilm Irvine Scientific. If I start talking about her illustrious career, it's probably going to take me an hour to describe, and I'm only given maybe a 30 to 40 seconds bracket here. So I can just say that she's been working as a senior embryologist from 2001 till now in various fertility clinics in Egypt, USA, and UK. And I'm going to let her, speak for herself, for her knowledge as she talks about what's new in vitrification media. Over to you, Dr. Manal.
Well, thank you very much for the introduction. So I would like first to thank everyone for attending and thank you for arranging this lovely event. So I will share my screen for. Share. Okay. So, right. So if you can see the screen, well, we can start. Okay. Right. So today we'll talk my presentation mainly about the new media of the vitrification and why we have this new media. So we'll talk about the formula, the new composition, how to use the media applications and protocol. And we will see how is the performance so far of this new media. And of course, we can't speak about media without talking about a cry device. So before we speak about the new vitrification media, I would love to give a short history about the original vitrification media from Irvine Scientific. So the original kit, what we call it the old version, which I don't like to call it really the old version, is still so far being used and still available for anyone who would like to use it. So it started in 2004 and it was the first commercial media available because before that, any embryologist would like to practice the vitrification. Usually they were homemade their media. So they were trying to compose their media in-house. So in 2004, this media came to the market to be the first ready-use media, and it's been since then 2004 being used till today. So in 2006, it's got the FDA clearance for especially blastocysts, but it actually covers all the stages. And in 2010, it had the CE mark so it can be used in Europe. And it's also used in Europe for all stages. And in 2016, it was officially the first media to be approved to use for oocyte vitrification, even though it's before that was used in that. And during these years from 2004 till 2018, there was a lot of media came to the market, and actually some of these media also adopted the same idea of composition of Irvine media. In 2019, just last year, Irvine launched the new vitrification media. So it's available in the market, especially in USA, for almost a year now, and it's being tested and it's used. So we can see from 2004 till today, the original kit being used successfully, and it has history of publications, all good results, and it's been used from 2004 till today. So it's a long years of experience and success. And it's not going to be discontinued from the company. It's available anyway, right? So let's move to talk about the new media and why the new media, what kind of advantages that it can bring to your lab. We know that we always have these advances in IVF, and we reach the point that we are all expert in vitrification. But also, there is a lot of events and advances recently happened in IVF. So in most of laboratories, we can see that most of the laboratories, they use the uninterrupted culture media. Because it's bring more stability to your lab. And the culture media from Irvine is called the CSCM, is the continuous single culture media. So, and also everyone use a manipulation or handling media in the lab. So in the recent manipulation media since 2012 is available, is the multi-purpose handling media. And it's based actually in a dual buffer. So the dual buffer is giving you a big advantage that you use less concentration of the buffer. That is a lot of, like, there was a period that a lot of embryologists, they didn't like to expose the gametes to buffer, because you have to stabilize your pH, you need to have a certain concentration of either heaps or mobs. But using dual buffer, you can use much less concentration of both, and yet you can have a secure environment and stable pH. So with the uninterrupted culture media and the handling purpose media, you can use only 2 media in the lab. And that is bringing you the idea of exposing your gametes and embryos to different kind of media during the process in the IVF. Now the vitrification media came to support the same idea, the new vitrification media, and it comes to bring the vitrification process in line with this idea. And it came also to convey all the advances in the modern IVF lab. So we know every vitrification media has a certain composition, but it's generally, you have to have a buffered basal media. So you need the buffer and basal media. And you need permeating cryoprotectant and non-permeating cryoprotectant. And also you can have macromolecules, which is, we use the protein. So in the basal media, we use the culture media as a basal media, and we need to buffer this media, right? We can't use it based on the sodium bicarb. So CSCM is our culture media, is actually used to be the basal media of the new generation of the vitrification media. And the buffer also is following the same of the multi-purpose handling media. So it's a dual buffer. It's heaps and mobs. And of course, we need to use a non-permeating cryoprotectant, which is very important in helping to stabilize the osmolarity, and also it helps to reduce the molarity of the permeating cryoprotectant. But we choose the triolose because it's actually has higher glass transition and help stabilizing the membrane, especially during the rehydration. And also triolose known as is good in glycosidic, they stabilize the glycosidic bond. And also it's interact with the water, so it reduces the ice crystal formation. So it has many advantages to use the triolose in your media. And also it has it's reduced stabilize the protein in the solutions. So with these advantages, we use this 3 differences in the formulation. But what is the complete formulation of this media? As we said, the basal media is a CSCM, it's a cultural media. The buffer is a dual buffer to give you more stable and secured environment. The protein is the macromolecule, the 20% DSS is a human serum albumin with dextran and gentamicin. And this media has no phenol red, right? So it's a clear media. Now with this basal media, we need to add our cryoprotectant so that it's basically been adopted from the original kit because it's a trusted formula and it was working since 2004 till today with a very good results. So we use the same concentration of the permeating cryoprotectant DMSO and ethylene glycol. So in the freezing solutions, we have two solutions, equilibration solution and vitrification solution. In the equilibration solution, we have 7.5% of DMSO and 7.5% of the ethylene glycol. In the vitrification solution, we have double this concentration, so it's a 15% of each. So the total permeating cryoprotectant is 30%, and also with 0.5 mole of trehalose. In the thawing solutions, we have three solutions: thawing solution, dilution solution, and washing solution. In the TS, we have 1 molar trehalose, and the dilution solution has 0.5 molar. And in the washing solution, there's no absolute cryoprotectant, non-permeating or permeating. It does not have any cryoprotectant, so it has only the basal media in the washing solution. So this is the composition, and it shows how is that supporting your embryos, that when you culture the embryos in the CSCM and you bring it to the vitrification media, there's no much change in the composition and the nutrients. Right, so how we use this media and how we apply it. For who was using the original kit, the protocol is almost the same. There's no change. So we have three protocols: one freezing for all embryo stages, and one freezing for oocytes, and one protocol for warming all stages, oocytes and embryos. So it's only three protocols. So we start with the protocol of embryo vitrification. And again, we have the ES and a VS for vitrification, so only two solutions. We start with the 50 microliter of the ES, then we place the embryo there and leave it for 8 to 10 minutes to equilibrate. And just before the time of equilibration finish, you dispense the 50 microliter of the VS and be ready. So when the time of equilibration all finish, you move your embryo from ES to the VS solution and leave it there for 30 seconds. In the first few seconds, you expect to see the embryo floating, and then you can bring it down. It's float again, you can bring it down again. Or the other option is, if you don't like to manipulate that much, leave it just to float for 10 seconds and bring it down to the bottom, and then it's not expected to float again. And after the 30 seconds, you can load your device and seal and plunge. Right, so it's quite simple protocol, 2 drops, right? And we are not dispensing the drop before, unless we use it. We're not preparing anything in advance, right? Right. So we move to the oocyte vitrification, and the oocyte is actually different from, there's a lot of differences in the oocytes from embryos. So we can't vitrify the oocytes in the same way we would vitrify the embryos. So we need to dehydrate the oocytes gradually. So that's the reason we use the washing solution, which included in the vitrification kit. And it's a 20 microliter of this washing solution. And then we also dispense 2 drops, 20 microliter each of the equilibration solution. And we bring the oocyte, place it in the washing solution for 1 minute, just to equilibrate to the new environment. And then we start merging. So we start merging and expose the oocyte to the cryoprotectant by merging the 2 drops and leave it with 2 minutes. After the 2 minutes, we merge the second ES drop to the other two connected drops. And also we leave the oocyte there for 2 seconds. And during this, 2 minutes. And during this time, we're going to dispense the third drop of the ES. Right. So after the 2 minutes, we move the oocytes from the connected drops to the pure ES and leave it there between 6 to 10 minutes. So the equilibration time in this drop is longer, during which we can see the oocytes shrink and expand again. Before the time of the ES in the equilibration third drop finish, we can dispense the 50 microliter of VS solution. And when we know the oocyte is ready and the equilibration time is finished, we can move from the third drop of ES to the VS drop and leave the oocyte there for 50 to 60 seconds. During which also again, the first few seconds, the oocyte will float. So we can pick it up from the top, place it down to the bottom, and we can place it at first 12 o'clock, then move it to the 3 o'clock, and then move it to 6 o'clock. And in this way, you'll be sure that it's totally rinsed and also being all surrounded with the pure VS, because this is the concentration very important to for glass transition during the vitrification. After the 50-60 seconds, you can load the device immediately and seal and plunge the device. So it's more steps for the oocytes, but it's necessary to understand that it's different from the embryos. Now we move to the warming protocol, right? So all the protocols should be in room temperature. And when we thaw the any specimen, oocytes and embryos, we have to start actually our warming from 37 degrees. So the reason why we use 1 ml of the TS solution to be warmed first at 37 degrees, and you use any dish of your preference, that's fine. And so 1 ml of the TS should be kept in 37 degrees. And when you are sure it's 37, you keep it warm in either non-CO2 incubator or a heat stage. When everything is ready, you have your straw, and the DS and washing solution also should be room temperature. Then in one dish, you need to dispense 50 microliter of the DS because there's no time to dispense this straw. And then everything is ready now. Straw is ready, DS is ready, TS is 37. Now we can start the warming by plunging the straw immediately to the TS after bringing the dish under the microscope, and then leave it there for 1 minute. After this 1 minute, we can move the specimen from the TS solution to the DS solution, and it's better to carry over volume with us. So when you carry your transfer pipette, you pick up the specimen oocyte or embryos, it's good to carry more solution with you TS and leave it inside the DS, which create the gentle warming, right? And leave it there for 4 minutes in the DS. During this time, you dispense 2 drops, 50 microliter each of the washing solution, right? After the 4 minutes in the DS, you move to the first drop of the washing solution. Again, you can repeat this step. You carry over with the volume from the DS solution to the washing solution and leave the specimen there for 4 minutes. After this 4 minutes, you move to the second drop of the washing solution, and that is the last step of warming, and you leave it there for 4 minutes. And after the 4 minutes, you're done with the protocol, you leave the specimen to recover in culture media. Right. So we've seen like 3 protocols, and it's all like all micro drops, it's a small volume. So how many applications that I can perform with each kit? So with the freezing kit, the whole kit can cover 60 applications from embryo freezing protocol. So if you're assuming that you're going to use only one embryo in one protocol, so that's 60 embryos. Or if you use more embryos in one protocol, then you increase the number. And for freezing oocytes, you can have 50 applications from this oocyte vitrification protocol. So again, usually we freeze oocytes together. We rarely somebody will in any lab that they freeze only one oocyte in one straw. So probably that is higher number for oocyte freezing, but for the application itself, it's only 50 application. And for worming kit, you can have like 12 application for worming. And that's of course for embryos and oocytes, right? So as I mentioned in the beginning, this media is available in, to be used since 2019, and it's FDA cleared and CE marked. But before anyone using this media, and since it's a new media, it should be validated first. So it's being validated internally by animal model first. So it's being tested by mouse embryos and hamster embryos and oocytes, and it's being tested against the performance of the old vitrification media or the original kit, because it's a trusted and we know how is the performance, and also is being validated and tested against other competitor media. But what I consider is more important is the human model. So what we have from the human model as a validation for this media. So thankfully for the World Egg Bank, they tested this media experimentally on oocytes, and it was good results for immediate survival, and after 2 hours post-warming, for the results for against the old version kit, and also against other competitors. So the recent study shows that this is actually a clinical application. It's been already used in one of the big centers. It's Houston Fertility Institute, a part of big group of IVF chain, and it's a big clinic, 4,000 cycles per year. They're using the old version kit for almost like 8 years, and they're very successful with this kit. But they decided to give a try for the new generation kit. So first they started to worm the embryos in the oocytes that's been already frozen with the older version of our kit, the vitrification kit, and they started to worm it in the VIT-NX, the next generation one. And then later, slowly, they introduced also the freezing and the warming by the new generation kit. And from these results, you see they tried with the big number of cases. There was a slight improvement in the implantation rate and the clinical pregnancy rate by using the new version or the next generation vitrification media. So that is, I consider this study is very important because it shows how is the performance, the clinical performance of this media. Right. So we finished everything about the media because we discussed the composition, the protocols, and what is the value of this new media. So we move now to talk about the device, and we are going to talk about the high security vitrification system. It's famous system, it's not new, and everyone knows this system, and it's very reliable system. So we have the HSV straws with the dedicated system, and of course with the opener, the Nepix to open the straws. But with the closer look to the straws, it's interesting to see how is this straws is advantage your practice. The straws coming in one kit like 3 pieces. The main piece is the capillary tube where you load your embryos, and it's transparent. It ended with preformed gutter, and the gutter is really nice to protect the loaded embryos or oocytes, right? And for easy handling, it's connected with colored rod. And this rod coming, it has like, it's available with 6 colors for better traceability and identification. The second piece is the long sleeve that is can be sealed, and the device is a closed device. But there is the end of this sleeve is pre-sealed already, and with a piece of metal to give weight for this straw. The other end is open with a wider opening like a funnel shape to provide better insertion for the main straw. And also there is the third piece that is the blue connector. It's making the insertion of the blue of the main straw to the outer sleeve easier. This straw was available long time ago, and it's being tested with oocytes and vitrification, with vitrification of oocytes and embryos. And it has also long history of experience with success. But also recently, with ISHRI guidelines, it's clearly stated that using the high security straws is recommended, especially with the COVID positive cases. So that was everything in my presentation. So we explain how is the advanced formula can serve to streamline your process in your lab with the culture media and with the handling media, and also based on the basal media, that is culture media, it should provide the optimum support for your embryos and the oocytes in your lab. And finally, there is a cross-swarming evidence that is being shown between the original kit and the new kit. And also the original kit has a very good results with cross-swarming with other competitor media. So I'm assuming that also the new generation media, we will see studies coming to see how is the cross-swarming with other media. So thank you very much for your attention, and I can receive any questions.
Yes. Thank you, Dr. Manal. That was a wonderful presentation. And you took us to all the years of various editions in the vitrification media. We got an insight in that. I don't think we have any questions here as such, but I just wanted to know, like in the oocyte vitrification, you have given that there was 6 to 10 minutes gap we have given for the oocyte vitrification. So what is we are looking at, like which oocytes would you end it at 6 minutes, and where would you follow it to 10 minutes there?
Right.
Because it's not like an expansion or something as blastocyst or this thing, right?
Yeah, sure. It's it's a very good question, and we have to respect all the practices. What does that mean? Not all of us have the same experience with the oocyte freezing, because I think we are much expert with embryo freezing. Like a lot of embryologists, they are much better experienced with embryos. But with oocytes, it's a little bit different. It depends on, like, if we took all the regions all over the world, there is some areas they are very expert with oocytes more than other areas. So we have to respect what is the difference of the practice. Then we have to accommodate all the practices. If you are very expert in oocyte freezing, like you've been working in egg bank before, well, you are the top expert. Then you totally know to understand what time, at what minute, I can move my oocytes from the ES to the VS, right? I know that this oocyte being totally equilibrated. And the description itself, I still consider it subjective. So the description is like, when your oocytes is like totally shrunk during the dehydration, and you have to see this oocyte go back by 80 to 90% of its volume. It's not going to be back to the same original status before you leave it in the ES, but it has to go like expanded again 80 to 90% of the volume of the original volume of the oocytes. So this is the time that it's ready to move. Now your question is, so what time we like 6 to 10 minutes is so wide range. Yes, it is wide range, but I personally recommend if one of the laboratories, they proceed with the oocyte freezing and they're not very expert with the oocytes, fix your time. So 10 minutes, it should cover all oocytes responses, because not every oocyte respond the same thing, the same time, the same, like they really behave differently in the ES, in the equilibration. Even if it's coming, all this group of oocytes coming from the same cohort of oocytes, like the same cycle of stimulation, they still behave differently. So you can leave it 10 minutes as a maximum time. So leave everything for 10 minutes if you're not expert, and then start to move. But expert embryologists, what they do, they put a group of oocytes and they just leave it in a different corners, and they know this, like this 2 or 3 oocytes, oh yeah, I know they are ready, so I pick them up, I freeze them, and then go back to take the other ones from the equilibration solution. But this kind of practice, it does not help embryologists that they have less experience in the oocyte freezing.
Okay. Thank you for answering that. There's a question here, like personally, which system would you prefer, an open or a closed system?
Right. So both of them should be working, but if you ask me, I used both of them, but I have to say that HSV straws is the best closed system. It's because it's very thin. It serves the vitrification very well, and it's fantastic closed device. But if you would like to use the open device, you can use the open device, but use it mainly for the oocytes, right? Because oocytes really sensitive, and we still don't know a lot about this like process. We still don't know a lot about how the oocyte responding and what is happening metabolically inside when they get warm again, and the torsion and the squeeze of the spindle during the dehydration and rehydration is something still needs study. So I know that we need a very high vitrification rate and very high warming rate when we are warming the oocytes. But I would like to mention again, if you're happy to use your closed device, be sure that your closed device is providing you with the very good vitrification rate.
Can I ask you another question here? When you're using shallow drops, what would be the like minimum volume we are looking at? Because I think in the embryo vitrification you have specified 50 microliters there, but in the oocyte vitrification, I think the ES1 and ES2, like what was the volume of the media there?
Right. So the media in the oocyte protocol, the first 1, 2, 3, 4 drops was 20 microliter.
Okay.
Right. And the VS always 50 microliter. So I won't say there is a minimum and a maximum. So that is the protocol that is being tested, tried, and is being proved that it's working very well with this volume, right? But it doesn't mean that you can't increase the volume. It's up to you. You can use as high volume as the whole vial for one protocol. It's up to you.
Dr. Manal, I don't think increasing the volume is what everyone is looking at. Decreasing the volume people would look at. Like, you know, how less a volume can we vitrify is like.
Right. It's, well, I have to say that I was using Irvine, by the way, since 2005. And one of the very good characteristics that you can have a very good results with saving. So when I was working in a lab, that is too much saving. It's fantastic. So you can go and if you would like to reduce the volume, I won't that much reduce the volume as you can have another trick that you can use more than one embryo in one protocol.
Okay, thank you. And there's another one question, sorry. In case of oocyte freezing, why do we need to put ES gradually on the top of BS? Why not just pick the oocyte in another droplet of ES?
I'm sorry, can you repeat the question again?
In the case of oocyte freezing, why do we need to put ES gradually on the top of BS? Why not just pick the oocyte and another droplet of ES? Like, why the merging of the ES and BS? I think that's a question being asked.
Oh, the merging is actually between the ES and the washing solution, right? Or handling media. So the washing solution should be the best media to be merged because it has the same composition, right? But in the older version protocol, we're using the handling media, any handling media that has buffer. So why we merging, why we have to do this? Because I mentioned before that the oocyte cannot be treated exactly like embryos. We need to dehydrate the oocyte gradually. Why? Because if you look at the surface in relation to the volume of the oocyte, it's a big cell, large volume, and less surface. If you compare this with embryos has a plenty of surface. So the cytoplasm itself, to be dehydrated, they have plenty of surface. So there's no problem and for the water to go out, right? And the cryoprotectant to get inside, right? For the oocytes, that might be very, very, very stressful. There's no much place in the surface to allow the water go out and the cryoprotectant to start to get in. We need to slow down this process as much as possible to give the oocyte some time to accommodate this. Hopefully this has answered the question.
Thank you, Dr. Manal. That was lovely for you to answer all the questions here. I think we are done with the questions. 1 second.
Saroj, can I ask a question?
Yes, yes, please, sir. Please go on.
Dr. Manal, that was a wonderful talk. You covered up almost everything and very nicely explained how the oocyte is different from the embryos and the original protocol of Mike Tucker that you explained here in freezing oocyte. My question is related to the composition of the media. The first thing is whether this next generation medium has any antioxidants in it.
Well, if you consider the protein as an antioxidant, which is work as that, it is. So we have 20%. That is high concentration, by the way, of the protein. And I always, I get by chance to read a paper with actually mouse embryos, it's not human. And it's very nice. It's a very old paper that when the embryologist wanted to discover the better concentration, what is the best concentration, what is the best temperature, what is the best concentration of protein, whether we need the protein or not, but that was the paper. So they found out that the 20% protein is very proper or the best concentration for this process, because the 10% was lower and it did not protect the osmolarity of the and stabilize the membrane as possible. And the 30% was very high concentration of protein that is actually conflicted with the cryoprotectant function. So the 20% protein, it should give you this function as an antioxidant.
Because sometimes it is confusing for especially the newcomers in the field, because in some references it is mentioned in percentage and in some it is mentioned as the mg per ml. So yeah, it's confusing, 10 mg per ml and 10%. So
yes, well, to be honest, it's confusing. And I was like, when I was I was reading about this more and more to get the idea, but when I was younger it was absolutely confusing and I did not get it for years, by the way. So yes, it's if you would like to express it by percentage, it's different if you express it by volume to volume or volume to weight, right? Okay. So if you would like to know what is exactly the percentage, right? So it's a 20% protein of dextran, that is a macromolecule, and the human serum albumin. So the human serum albumin concentration is around 10%. So the total is 20%. Okay. It's around, I think, 10 milligram per ml, if I'm not mistaken.
Yeah. So another question regarding the composition was like the change in this next generation kit is that in the equilibrium solution, they have used the CSCM.
Right. Okay. So it's not only in the equilibration solution, because you know that we need something, what we call it the solvent, right, to start to add the concentration of the cryoprotectant. So the solvent or the base that we change the concentration of the cryoprotectant to make ES or to make VS or to make TS, it's all based on the culture media. So we have a culture media that it has higher buffer, and the buffer is dual buffer, right? So that is the basal media. It's also all contain the 20% DSS and the gentamicin. So that is the basal media where we take this solvent and start to add different concentration of the cryoprotectant, whether permeable or non-permeable or both together, and create the different solutions.
Okay, so the difference is that because most of the vitrification media, they contain the heaps or MOPS buffer or maybe dual buffer, but in addition to that, it has the culture media as well.
I am not sure that I know any other media that has cultural media, but
no, not the other media, but this one.
Yeah, they have basal media, but up to my knowledge, I know that the CSCM is being designed mainly for human embryos, support human embryos. But the other basal media was available. It was actually based on also animal model more than human model, but it was supporting also human model, but it did not being designed basically or originally for human only. So CSCM was actually from the start genuinely being designed for human embryos, to support the human embryo development.
Okay, so can I ask a trade question, a trade secret question? So what is the concentration of CSCM in the?
Oh, I can't say what is the concentration exactly, because it's okay. It's a manufacturer. No, it's not secret. It's a manufacturer thing. And okay. I know that well, I had this this before. I have the experience of making my own media in the lab. So I didn't know if every embryologist, like especially young embryologists, they didn't do that. But if you get the experience and to know how you can make your own media in the lab, so you know that there is a solvent and you have a certain ml and you have your own calculation to calculate, all right, so I'm going to make this percentage, I'm going to make this percentage, and you have to have a scale and weight, and you know how many, and you start to make your own media, right? So the CSCM itself, it's there, right? And all the different, and also the buffer is there. So I know that the buffer, it has to be the main buffer system. It's not the bicarb system. So our culture media, CSCM, the main buffer system is the sodium bicarb because it's a culture media. But when we start to make it for the vitrification, I'm sure it's the same composition, right? But the dominant buffer is actually the dual buffer heaps and MOPS, right?
It has to be,
and,
yeah.
Yes, but the concentration is actually suitable to give you exactly the percentages of the concentration of the permeating cryoprotectant and non-permeating cryoprotectant.
All right, thank you.
Thank you, Dr. Manal. That was lovely, and lovely the way you answered all the questions. But I think we are quite late for the next session. So I would hand over.
Thank you very much.
Thank you. Thank you, Dr. Manal.
Thank you,
Dr. Manal, and thank you, Dr. Saroj, for moderating a really good session.
You,
and I would like to carry forward the session. I would like to invite Dr. Rahul Sen to introduce our moderator and take forward the session.
Thank you, Dr. Akash. Can you please share the slides? Yes. So it gives me a great pleasure to welcome you all in this Pre-Symposium Masterclass of embART-2020. So moving on with the next session, which is a panel discussion. And for that, I would like to introduce the moderators of the session. The first one to come up is Dr. Sujatha Ramakrishnan. She's head embryology at Nova IVF Fertility. She is a PhD in biochemistry and molecular biology from University of Mysore. American Board of Bioanalysts certified embryology lab director. And the next one to come up is Dr. Krishna Chaitanya. Dr. Krishna Chaitanya is currently a scientific director at Oasis Center for Reproductive Medicine, Hyderabad. He's a Master of Clinical Embryology from Monash University in 2008, postgraduate diploma in reproductive sciences, and he has completed his MBBS from Rajiv Gandhi University, Bangalore in 2005. So I welcome both the moderators and please carry forward the session. Thank you.
Thank you very much, Dr. Rahul.
Thank you so much, Rahul, for that introduction. And without any further delay, let us start on with this wonderful topic today, adopting new technology in the laboratory. Joining me in today's panel are our experts, Dr. Pranay Ghosh from Delhi, Dr. Sanket Dhumal from Mangalore, Dr. Keshav Malhotra from Agra, Dr. Sayali Kandhari from Mumbai, and Dr. Karo Kant from Delhi. So they're going to be our expert panels who are going to enlighten us and help us. How do we go about with this whole aspect of adopting the new technology? So, way back, I think in the late 80s, Joyce Harper and Alan Handyside, did the embryo biopsy in the first PGD, and that was a breakthrough, a very important milestone in the history of IVF. And subsequently, we went ahead and came up with multiple versions of pre-implantation genetic testing. Until a few months back, the STAR trial came up with its final inference saying that PGTA or PGS, wherein you're testing it for aneuploidy, doesn't seem to be improving the live birth rates. It almost took 3 to 4 decades where people have done research, they've clinically offered this to a lot of patients. And finally, a large RCT, after 4 decades, finally says there's absolutely no value addition with this wonderful technology. Similarly, again, almost 3 to 4 decades back, when ICSI, which was, all of us know that it was an accidental discovery in the field of ART. I'm sure it didn't go through the regular way of validating the newer technology. It is a blessing in disguise for, of course, severe male factor infertility, but of late, I'm sure more than 80% of clinics all over the world are actually using ICSI for all the couples who are seeking ART. Surprisingly, I don't think the amount of RCTs or meta-analysis or the scrutiny that's required to accept a technology did not really happen. So, ladies and gentlemen, so let's now have a panel discussion where my expert panels are there who are going to enlighten us about, one, the new technology that we already have in 2020 in our laboratories, and all those technology that we could actually access. What are those little tips and tricks before we could even consider adopting a new technology? And after you've adopted it, how do we go about with it? Along with Dr. Sujata Ramakrishnan, I'm the co-panelist, Dr. Krishna, who's going to be moderating this session. All right. So myself and Dr. Sujata have absolutely got no conflict of interest. Unfortunately, no company came forward to sponsor anything to me and Dr. Sujata to do this moderation. And also to disclaimer, none of the panelists have threatened me and Dr. Sujata to do this panel or influenced us to ask these questions. So that's Joan Purdy in way back 1970s with Bob Edwards with their age-old lab where I think there was hardly any technology. Most of the things that they were doing were manual or semi-automated. In 4 decades, see what we've all done. We've got a state-of-art, high-tech, wonderful technologies. There's n number of technologies, gadgets, electronic gadgets, computers. I think it's got quite complicated out there. And this is what was my feeling when Ved told me that this is the panel and that I'm going to do. And when I just sat down to see what has been the progression with technology, and I was totally blocked and confused. And this is, I'm sure, the situation with most of us in the laboratory. Every conference happens, ESHRE or SRM, we've got the clinical team running to the embryology team telling us, we saw that, can we just get this? We saw a new set of media, can we use this because we probably have a pregnancy rate of only 40%. And I know we are all going haywire and confused. And this is where my expert panel today is going to throw a lot of light. So to all my panelists out there, what is the first thing that comes to your mind when we're talking about new technology in specific to the field of ARC? So Keshav, I don't know if you are okay to take that question first.
So a very good evening to everyone. And first of all, thanks to Ved sir for inviting me here. And thank you to both of you for moderating this wonderful panel. I think for, if I talk about new technology and if I talk about as a group, I think the most important thing that we feel as a group is that this technology should benefit our patients. It should be something new. It should set us apart from the other clinics. And like, these are a few things that we really feel. Now, we don't really consider them to be marketing strategies. We don't, we would look at technologies and like try and figure out whether this could actually benefit the subset of patients that come into our clinic. Because if we talk about
Keshav, I'll interrupt you there because I'll come back because we will get to that whole little checklist that we would want with the new technology. But one thought when I'm saying new technology in ART, let's say when the management of Rainbow Hospital said, we want you to look at RI witnessing system at Rainbow. What was the first thing that came to you? What did you think this was more of a marketing strategy that the whole management wanted to bring this in? Or do you think this is something very novel, or this is going to do a game-changing thing in your practice?
Yeah. So actually, it was the opposite way around, where I actually told the clinic and the board of directors that this is the technology that we should look into. And the sole purpose of utilizing this technology or incorporating this technology was the fact that all of us travel quite a lot because we have 10 centers. So we are frequently visiting all of these centers. So to have that mental peace that once you're gone, once you're outside the clinic, and work still goes on because there's a team which stays back as well. So just that mental peace for us that whatever is going to happen is going to be documented properly, is going to be recorded, etc. That was the only thought behind it.
So Sayali, when the thought of time-lapse came at your center, a new technology, what was going in your mind?
So good evening, everybody. It's lovely to be here. And again, thank you for the invite to be here on the panel. So Dr. Krishna, when it came to time-lapse, the way Dr. Keshav said, it was more of the, more of me telling the team that we need to get time-lapse in, because this was something that we wanted to get involved with in research or in looking at how it really changes the entire workflow. The way it was said, you can't be in the center all the time if you are traveling.
So it was more of being novel and a game changer is what was your take?
It was basically something that would improve the workflow for the entire laboratory, because I would be able to check and annotate embryos when I was not on the spot.
So
I was training my team for about a year to do that. So,
wonderful, Sarah. So Pranay, I'll kind of put you in a fix here, Pranay, because I know you're a clinician and an embryologist. And I'm sure you're kind of balancing the management, the clinical team, and the embryology team out there at Elixir. So what is your take? Were there been any situations where you thought this is more of a marketing strategy and this technology is not going to be of much of use for you in the laboratory?
Thank you, Dr. Krishna. I hope you can hear me loud and clear.
Yes, yes.
Yeah. So, I would, amongst all of the panelists, I would want to be old school. And since I do carry some of my clinical years behind me, I would want anything to be incorporated into the clinical workflow or the laboratory workflow to be evidence-based. And I accept that you cannot have evidence without trying it out on patients and generating data, and then evidence does come later on. But I believe that we have a responsibility to our patients, and I see a lot of words like precision medicine and personalized medicine being thrown around, but we need to be very clear that what are we wanting to achieve? Are we wanting to achieve increase in live birth rates, decrease the time to pregnancy, decrease miscarriages, improve documentation, reduce mishaps like mix-ups, be medical legally safe? If and when a technology fulfills most of the check boxes for these, I probably would jump upon it. I would want to incorporate it, but I should also take into account the hidden costs. Would I, like I debated it amply with Keshav on a couple of occasions as well, where does the cost factor in? Are we passing it on to the patients? Because it would not be a one-time cost.
Wonderful. That's always a huge challenge for all of us in an Indian scenario. But I think down the line we will probably balance it out in our subsequent discussions. So Gaurav and Sanket, would you want to add anything? The first
Krishna, maybe
yes.
We could ask them what do they think about technology being a tool to standardize or to reduce subjectivity in the lab. Do they think that way?
Yeah, Gaurav and Sanket, if you could take that, please. I'm hoping Gaurav has joined us in Sanket.
Are they connected?
Yeah, I'm here.
Hi.
Hello?
Yes, we can hear you, Sanket. Your comments on this, Sanket, if you've heard the question right.
Sanket, my question was, okay, apart from the technology, like a tool to improve pregnancy rates or improve the outcome, or as a, whatever is mentioned here on the slide, do you people think that it can actually help you in the lab to reduce subjectivity, to kind of streamline the activities in the lab, to bring some kind of a standardization?
Yes, of course. As there are some techniques like time-lapse and witnessing systems, so which will standardize and which will reduce the burden on the operator, so that things will be streamlined in much better way when compared to manual operators.
Wonderful. So Gaurav, are you there? One last comment on this. Yeah. When we are thinking of new technology, what is your take on this?
So, first of all, good evening, everyone.
Thank you, Gaurav, for joining.
So whenever I look for new technology, I always look for problems first of all, because before finding a solution, you should have a problem in your lab, right? Like when I add the microfluidics in my lab, so I was seeing that although the semen sample was okay, patients are having high DNA fragmentation. I have no solution with me so that I can apply and improve the results. I found this study, as Dr. Pranay said, that you should have the multi-centric studies with you. You should have the clinical, assess the clinical and the cost effectiveness of the technique, whatever you want to apply. Then I used it in my lab and I found a very good result when I applied microfluidics particularly. So yes, it can help you. You have to assess it. You have to ask your friends as well. Like me and Dr. Pranay, we discuss a lot whenever we have to add a new technique or we're going to do something new. Like how is working in your lab. It is very important that we communicate as well with our friends.
So wonderful that, all of us are open for changes, open for new things. I think that is what has led to this wonderful journey of ART in the last 4 decades. I think we've gone from birth of Louis Brown to editing the gene. Well, long way to go with gene editing still. Our success went on from 6% to almost 60%. But unfortunately, failure is still a factor of fear for all of us in the laboratories. Multiple pregnancies is still a challenge. Prediction is still an enigma. And this is where I think there is a huge scope for newer technology to be adopted in all our laboratories. So now, to all my panelists out there, we come to those little checklist that you're going to do. Take any technology to that matter. How do you get started? Like Keshav and Sayali said, it was more of they initiating the thought and getting the management to invest in the technology. Whether it's the management clinicians or we as laboratory people. How is there a basic equation, a set of questions that we got to be doing homework on, and then we could adopt this technology? So, Sanket, why don't you
Can I
take?
take
Let's
that?
just one point so that we could get all the panelists commenting on that. Where do you want to get started?
Okay. Yeah. First thing is, if there is a demand, we have to see that. After that, we have to see for the safety and effectiveness.
So
and
first
cost
is
effectiveness.
demand. So Sanket says, so the first thing is you would want to see if there's a need to even really adopt or bring in a newer technology. Because, there's so many times we have this, I've had a lot of times where, the clinical team is looking at us and saying, do you really think we should change the media? And I'm like, when you're really having a decent performance, a bigger thing of changing the media overnight is something that I might not really be happy doing it as an embryologist. So I think the first step is to identify if there is a particular need. So Gaurav, what would be the next step once you've said, okay, probably I need to bring in some technology? What is the next point? Gaurav, if you're there.
Has to unmute.
Yeah, second thing is definitely safety. I want to read about what is the impact of using the technology on the
All right.
Yes.
So
Gaurav,
yeah.
Now how do you ensure safety? What plans? Just give us an example of any technology which you want to incorporate in your lab, and how do you envisage the safety of that particular technology?
Like madam, I can talk about some medias. There are recent studies that if you culture in certain media, you have a large offspring disorders, right? So we shifting to that media is not that good. So we can have a media which is giving a better result and it is not impacting the outcome.
Wonderful.
I don't want to name the media right here, but we all know it. We have studied, read about the particular study which I'm talking about.
So it's the need, it's the safety. Keshav, what's the next point?
I would also look at like what are our skill sets and how the technology is going to complement your own skill set. So
it's adapting and training and the skill matrix.
Yeah.
Wonderful. So, Sayali, what is your next point that if you want to add there?
So we have this excellent points that we should need, there should be a demand, there should be safety, we should look at our skill set. And I would add that we need to really look at the whole process of adopting new technology like a research experiment. We need to know what our aim is.
So evidence-based medicine, the literature. Okay. So Pranay, what is
we
it
need
that
to
we
do
missed
the
on?
review and
yeah, I would complete the list, but I would want to say that what I feel is most important is evidence-based. I mean, it shouldn't be something like endometrial scratch, or it shouldn't be something like MZ that years and decades of practice, and then you tell the patient that, I'm sorry, we thought that it was very good at that point of time. We know better now, so we'll do it better for you.
I think that's what happened with PGTA.
Yeah.
Everybody went gaga about it. People thought, they didn't have the right skill if they were not able to biopsy the embryos. And now the evidence says it probably doesn't help.
Yeah.
So probably just to complete the list, the cost effectiveness, because we are working in an Indian scenario. So I'm pretty sure time lapse is a pretty benign, innocuous thing, which has now proven to be safe for our patients. Whether or not it actually improves anything is yet to be debated.
Proven.
But in the West, they have streamlined it into their laboratory flow without passing a considerable cost to the patient. Can we do that in our settings? So we really don't know, because we really have to factor in the cost in Indian scenario. So I think just to complete the list.
Wonderful, wonderful. I think so. All the expert panelists out there, just help me out to see if we've missed out something. And if you are wanting to change the chronology of the way I would put it. So for me, it's all about to first audit my data, look at the KPIs, benchmark my performance. And that's what is going to exactly tell me is there a problem, a need, a need to adopt or bring in something new? Then what are the options? Let's say my clinician says, you know what, our pregnancy rates are 40%. Can we go to 60%? Now, what are the options that I would want to explore to go to that point of 60%? And that's what I would want. So what are those technologies? And when I'm saying technology, what is that I would want to look in? Well, people said safety, people said evidence. So we'll come there. Then I'm sure there's a particular equation, a systematic way of bringing in that technology. I just can't be buying a set of media and tomorrow morning put it across to all my patients. I'm sure that is not something that we all would want to do. And this question, I think, would all of us be happy to inform our patients that we're doing something new? Sally, like a journalist, a very shrewd question. Did you really inform your patients the time lapse was a newer technology? And we didn't really have the right evidence, but we're still putting them in a time lapse incubator.
Okay, so regarding the counseling part, so I would take this as what I would counsel the patients before they go for time lapse. So we used to have a counseling session with every patient before they went for time lapse. And the main part of the counseling was regarding the fact that we would be observing the embryos in real time. We didn't exactly tell them, we told them that we don't know if this improves results. So that was very clear from my side, at least. Unfortunately, the clinical team is not always on the same page as the embryologist, but it was clear from our side that we don't know if this improves pregnancy rates. We are adding this technology because we have had issues with your particular cycle, and we would like to try uninterrupted culture, which we had some evidence for at that time, and which was not possible in our other culture system. So that was something that.
So every panelist out there agree with Sayali that it's a basic right of the patient to be informed that we are trying to try something that really has not been validated at this point in time. Any conflict of thoughts at that from any of my panelists?
Not really, but
I don't support Dr. Yeah, Keshav, go ahead.
So if you look at the evidence now, you will find good quality evidence for, like, you'll find benefits and you'll find pros and cons. So it's very easy for any clinic now to justify their use of technology, because they can just quote one paper which says, yes, this tells me that this technology is showing good results.
Sure. Yeah.
So I don't think that like that should be the critical aspect. Now you have to judge the technology based on your subset of patients, and I think that is where all of us lack, because none of these technologies have been tried and tested so robustly for the Indian subset of patients. And that's something that I think all of us have to work on. So maybe we will probably add one more point of data collection and reporting into this flow chart. And
I think see, another thing that you can decide upon is how invasive is this technology. For example, if you are having a witnessing system, it's not going to harm the patient in any way. So, like that is different, but that.
Yeah. Sanket, you wanted to add something?
Yeah, sir. So the thing is, even when the ICSI was introduced initially, hardly there was any evidence that supports the effectiveness and safety of this technique. But it picked up robustly. Now you know what is the fate of the ICSI in day-to-day life. So I think even with less validation also we can go ahead, because if it is maximizing the success rate.
I beg to differ.
Sorry, Pranay.
All right, maximizing success rate is a very, very big.
There are situations where you compare ICSI to IVF, and then there are situations where I can say that a stage 4 patient, terminally ill malignancy patient, would benefit from a drug that hasn't passed through a phase 3 trial, and that is his only option. So we are not going to wait for evidence there, because that's his only option. Similarly, failure of fertilization with IVF.
That means it's taking a risk.
Yeah. I mean, I'm saying that you are trying to compare apples with oranges. You cannot have. There is no way of having assured fertilization apart from ICSI for oligospermic patients or azoospermic
Wonderful.
patients.
So let's not get very technical with a lot of techniques out there. I think there's a lot more to discuss there. I think all my experts agree that this can be a basic formula the moment each one of us is thinking of a newer technology to adopt in our clinics.
Yeah.
Can this be a basic model where we kind of have a little checklist and we go ahead?
So, Dr. Krishna, I agree with the fact that you should begin with if there's a problem and then go to what technology you want to look at to solve that problem. But then there is this very open-ended step that what are your steps to adopt? Now, just from one personal experience that when we were, I'm not talking about time lapse, I'm talking about the fact that 10 years back when I was starting the egg bank, and had to begin oocyte vitrification with hardly no information in the Indian market or any seniors who had a lot of experience. The first thing that we had to do was kind of get the training to actually do vitrification. So it's not exactly getting the tech in the lab and starting to do it like Dr. Keshav said. You really need to have the skill set before you even offer it to patients. You need to be able to do it on discarded. So the same thing with time lapse. When we put it in, we did not actually start with embryos that were healthy and live. We wanted to first check whether it was really adopted in our workflow. You know, workflow really gets changed when you put any new tech in your lab. It may be witness, it might be something as simple as blastocyst culture for neo labs. So the steps to adopt, I think there should be a way of getting in professional help to help that particular laboratory to get the new tech in. And this is where
the learning curve. I think the learning curve was what.
So the vendors are of use, seniors are of are of immense help here. So the
this
embryology.
So can we just put it into these 3 points? I think this is from Joyce Harper's paper way back in 2015, where she wonderfully talks about how to implement a newer technology. So she says efficacy. Whenever you're picking something, look at the efficacy. It could be anything. It could be right from, is it just to the patient or to the clinic, or it's more helping for the scientific people. Safety, of course, is important. Effectiveness, the risk-benefit ratio, the cost implications, all of those things. So I think 3 essential aspects that she says cannot be overlooked with whatever new. Whether, when we're talking about media or incubator, I think that's more a straighter way of impacting the gametes and the embryo development. But, on the other hand, you're also talking about certain technologies like a pH meter, which is more automated. I think just yesterday I saw Alex putting a VOC meter which just sends the air from one of the Honeywell companies. I think we also have that which probably are not really impacting the dynamics of the culture system. So but I think if you're looking at these 3 things, probably it would give us a lot of information and it would guide us in making the right decision. And this is Joyce Harper's paper showing the whole paradigm of this is actually how you should be getting in technology into your practice, where you first have a hypothesis-driven research where you're trying to look at the basic science, you're developing a design, you're then doing animal experiments, then you're taking some donated gametes and embryos and you're trying to do that. You then do a little pilot study, then a multi-centric, look at the cost effectiveness, and then it should do. And sometimes it should take an entire life of a scientist, and still we probably have not ended the research. So that is where I think ICSI took off in a different direction, and things like metabolomics or genomics or the whole omics are still at a very primitive level of research. So what we will now do is we will pick each segment of our day-to-day practice, quality management, the sperm, oocyte, embryo, implantation. Look at the technology. And I want you people to tell us if there's something new that I've missed out, and where are we? Do we adopt it, or still do you think there's a word of caution for all my audience who are eagerly looking for your expert inputs? So when we're talking about total quality management systems, I think if I'm really talking of what's new, I think the whole automation of the lab in terms of having an automatic information about temperature change or osmolarity changing, and the incubator having fluctuations. That's where we have actually progressed in the last few decades. Anything else new that has come up into this field of monitoring your equipment?
Cryo tank management is there, which is
yeah. So
new.
I think that those little
and very important as well.
But one question, Keshav, I really get amazed when I look at these little gadgets trying to tell us that everything is automated. You just can go home and happily sleep. There are times where I feel, what? There's a lot of gadgets and machines in between me. I'm just interpreting what the machine's interpreting, and is that right?
Okay, so it's a tough question in the sense that because we as embryologists sometimes don't have that kind of a knowledge about like the physics or the chemistry of what goes on in an IVF laboratory, and that's where I think a trust element comes into play. But again, we here struggle where, especially in the Indian scenario, we struggle whether we trust the Indian vendors or we trust the foreign vendors, and automatically our thought process is like, ye foreign hai to ye acha hoega. Right. So
and there's so many times it's been 13 years in the laboratory, and still there are times where the moment I look at my media, it's kind of slightly more pinkish than what it normally looks. I put in the pH probe. I have a bedside blood gas analyzer, which takes hardly any media, is so quick to read, and the pH comes absolutely fine, 7.3. But my clinical acumen, when I look at the color of the media, I just don't like it.
Yeah.
And I'm not really happy with my blastocyst conversion. So my clinical experience says no, probably that media could be the problem. But my gadget is saying no, your CO2 of your incubator is fine, your pH is fine.
I'll always trust my clinical experience over, like that comes over anything else.
So where is the technology coming here?
Machine, right?
Technology in this scenario is coming just to aid you, like in times where you're doubting yourself, right? So I think that's where, and especially in certain scenarios, like I was talking about cryo tank management. Now, in here, my clinical experience is of no value because I don't know whether the tank is going to fail or whether the nitrogen is at the right level or not. It's a tedious process, and most of us will just refill every 3 to 4 days. We'll not even bother about leaks, etc. We won't even be able to find out in most situations, and that's the kind of situation where I would trust a gadget over my staff or myself.
Pranay and Gaurav, any input from your end?
Yeah, just make it short. I think it's also who's using the gadget. If it's Kesha or if it's just random somebody. So a fool with a tool is still a fool. So I think there's no better way to put.
I love those idioms. From where you find them?
I think he has them written in a notebook somewhere.
That's what I do is tell you how come to pay that much here. So I think it goes, I think if you are an embryologist like any of us in the panel or the moderators, you would be anal enough to have at least a couple of CO2 analyzers and just be double checking, triple checking everything. So I think that's one place where I don't have any qualms about spending on technology. I would
Krishna, I think, like especially in quality management, technology would definitely help because it would pick up things which are elusive to human eyes. See, as you said about the pH, okay, like if your pH meter, and especially such a sensitive one, is showing a correct pH, then I think, you have to rely on that.
Wonderful.
See.
So I think Gaurav and Sanket, one last pointer as to, as Madam was saying, Sujata Madam, that it's all about the preciseness, this whole automation and it's bringing in. What is your take? What has been your experiences?
So can I answer?
Yep.
Yeah. So looking at technology now, we started IVF from a desiccator. Right? Now we're having a new technologies, new incubator which tells you everything. So for me, technology is something which, for TQM, is something which reduces my stress in the lab. Like earlier we had only pyrite kit to take the pH, right? Which were not that much reliable. But nowadays we have CO2 analyzer, we are moving to pH. Now we can have readings in our mobile, we can see through apps. So it is something which gives me more confidence in the lab. That whatever I'm doing is better now.
Wonderful.
Convenience.
So absolutely, Keshav. So the last question to close on this slide would be to Keshav. Since we all know that you kind of started the RI Witness first in your laboratory. Well, all of us are sure that this is a wonderful technology. It just makes life of us as embryologists, more easy because you know that you've not mixed up samples. But as a beginning, I'm sure there are challenges. Probably there are delays in the dish screening it. What were those challenges that you've had, and do you really think we should still be going ahead with this technology?
Okay, so I'll tell you, I really didn't face any challenges as far as the time management is concerned. So there weren't any delays in screening of the dish or the reader not reading it appropriately, etc., etc. And that's where I chose RI as a system over the others, because I tried and tested all of them, and this is what I was most comfortable with. The only delay that I would actually say was like getting used to the flow of work now, because certain SOPs were changed, especially like egg donation cycles, etc. You needed to add like certain extra checkpoints which were not there before. So just getting used to those checkpoints initially for the first few months, I think that was the only challenge for us. And
So there has been a significant little learning curve challenges, which I'm sure these are teething issues, and one gets used to it. So
and
I think
most of these, I'll just add one more point here. What most of these new, especially with the witnessing systems, you always have to like try them out in their trial mode first, at least for 1 or 2 months, and then only switch it on to your normal mode, so that if there are any mismatches which are occurring due to the problems with the flow, at least those you can still be sure that it's in the trial mode and it's not being recorded initially. And once you are all trained, all of your staff is trained, you can switch it on to your normal mode, and after that you can't go back.
So all the panelists
Krishna,
give us
I have a question to ask to Keshav.
Yes, ma'am.
So Keshav, with this witnessing system, did it really help the reduction of tedious repetitive tasks in the laboratory? Because like you just double check, you check this, you check things on the paper. At least you could reduce the number of such.
I still do it.
Okay.
Honestly speaking, I still have my checklist. Whenever we start a procedure, we have a checklist for each procedure. What the witnessing system adds to that is that all of this is documented electronically. All of this has so your electronic signatures are there. So with the normal checklist, it can still be filled wrong. It can still be filled, like if an embryologist is doing, you can still fill in the wrong name, etc., etc. So it can go wrong.
So did it increase your workload?
I don't think so. It didn't increase our workload because it really incorporates into the normal SOPs of the laboratory very easily. And only thing that you need to add into your workflow is that you just have to click once or twice on the tab that's there. That's all. So it really doesn't change your workflow. It just improves and makes it more robust.
So all these newer developments, newer technologies that have come in the segment of TQMS is taking us in the right direction. Is that the right statement that I could make if I'm allowed by my panelists today?
I think it's the right statement.
Yeah, it's right.
So we all are all set and good to adopt and embrace this new gadgets, which are probably making more sense. I'm sure it is still a game of obsession.
So it depends on the gadget though.
Dr. Krishna, I have to make a comment. Funny that you mentioned embrace. So another technology that we are just jumping upon before validating.
I love controversies.
Yeah. Can I add one more point here?
So
please.
So whenever we're thinking of adapting a quality management system or any technology which is improving the quality of the laboratory, we all have to think that this is something that we're doing for our own labs, and these costs should not translate to the patient. So that's something that always has to be incorporated in your capex budget, and that's the way to go about it. And then I think you are completely justified in adopting any technologies.
Wonderful. So coming to cryopreservation, I'm sure all of us and the evidence also supports vitrification as a mode of cryopreservation in 2020. No, there are wonderful TQMS gadgets which are helping us monitor the time. You know, we just had a wonderful lecture before this about how do we optimize the whole system of vitrification. You know, when we're kind of sure of a lot of things vitrification is doing. Is there a need to automate this new technology? Well, I'm sorry, I've got nothing against Gavi, but the whole point of automating it, are we really doing it? Is this the right move?
So I think this technology of automated vitrification is quite useful if you are an egg bank or somebody who's doing this procedure day in and day out, and you just don't have enough personnel, or it just makes more sense economically to put a machine to do this. However, I do not think currently the specifications of, there are multiple machines like this in the market. This is not the only
So
one.
wonderful example, Dr. Sayadi, you put across.
Yeah.
You know, when you take a technology, I think most of us as beginners, you know, for us, we're just thinking in one direction. You know, we're just looking at it at that one step. Is automation needed? But why is it needed, and where is the longer application? So if you're really having a huge, large-scale program where there is going to be an element of human error and human fatigue, I'm sure a machine like Gavi absolutely has got a wonderful role to play. So that is what I ideally wanted our panelists to comment on. You know, it's all about balancing it out. You know, with whatever technology you take, you know, it could be the iPads, the iPhone, or little things in our kitchens. I'm sure each one have got a different perspective to look at it. And we have our own explanations, reasoning to adopt and embrace it. So anything new that has come in the field of cryopreservation that you want to enlighten us?
Can I say something?
Yes, please, Gaurav.
Can I comment something?
Please.
So it is about Gavi, making automated vitrification. So I don't like anything which replaces a human in the lab. So we are in a country where we are looking for jobs, right? So we have to balance that as well. That we are giving some money or some financial support to a person. We should not replace it with a machine. We're in a scenario where we need more jobs, more money.
So I'll hold on with that thought of yours, because that's my probably fourth slide from here, where we all have to debate whether it's man versus the machine. I'll come back to you there. But when we're talking about cryopreservation, anything positive or limitations the technology has challenged us in the year 2020?
Yeah, can I comment something?
Yes, please, sir.
Yeah. During this pandemic situation, there was a huge demand for liquid nitrogen, this thing, because there was shutdown of few plants all over the country and all over the state. So there was a huge demand, and there was, see, if you see the picture there in the display, you can see a lot of manifolds there, which are connected to the storage cylinders. So that automatically refills the liquid nitrogen levels. That ensures that the levels are maintained properly instead of overflowing the tanks with the excess like liquid nitrogen. So it is like very precise. So I think technology really helps in this kind of situations when there is shortage of raw material.
That's wonderful to hear, Saket. So coming to the embryology, I think when it comes to sperm, there's so much that research has invested its time and energy in the ovary, oocyte, hormones. But when it comes to the sperm, I think काफी सारा इंसाफी हुआ है. Panelists, please speak out. Are we in the right frame of technology? Are we moving in the right direction? Because, you know, so many panels I attend, there are so many gadgets they flash on the screen. At the end, I'm not able to take home one answer and say, look what I can tell my clinician, I'm going to do this, and that's going to solve my problems of male factor infertility. Where are we right now?
I think,
So,
Krishna, maybe you can start with Max, Krishna. Maybe you can start with your experience on Max, and then we will let everybody, talk about their experience with technology in andrology.
My experience with Max, with the RCT I'm doing, I didn't really see a benefit. And or in general, rather than pointing at a technology right now, my options for patients less than 5 million sperm count or the severe OATs, I don't have any option because microfluidics Max doesn't really help for them in terms of sperm selection. Neither Pixie does that. The only option I've got is MC, but I don't think it really made a difference. So I'm not left with an option for these less than 5 million sector, but I would want to learn more from the panelists today.
So, Dr. Keshav, sorry, Dr. Krishna.
Yep.
Yeah. So there are different things are available. Like we have Pixie, we have MC, we have microfluidics. So these are not created overnight. There are lots of hard work being done by lots of researchers. Then they've been created. I say not just jump up and say MC is not beneficial. So MC is beneficial when you have teratozoospermia, right? So when you talk about Pixie, Pixie is good. When you have higher miscarriages rate, then you can try Pixie. Like if I have a patient who have a good sample and has a high DNA fragmentation, then I can go for microfluidics. So we have to
Gaurav, in medicine, I don't think logic actually, logically it's right. When you have missed abortion, you have to use something like Pixie. That is logically right. But then that does not always give the right kind of outcome in medicine. Pranay, do you want to comment on this?
It comes down to evidence. I mean, you look at the evidence, you would say that Cochrane doesn't have any substantial final word on any of these things. But again, when he says that it is not that they have come up with it in a day, so have the researchers who are compiling the evidence as well. They have looked at RCTs and meta-analysis and compiled all the data across the years. And this is the MC data is over decades now.
So the thing is, when you are coming up with a technology, most of the times you start with the logic. You think that, okay, in teratozoospermia, when you have a technology like this, it helps you to identify a normal sperm. But then whether that gets translated into actual results in a clinical scenario is $1 million question.
Yeah, that helps actually. See, I know a person, I know Dr. Geeta Goswami, she does lots of MC cycles. I don't have an MC with me, but she does lots of. And I asked her, what do you do in OATS? She said, I do OATS, MC, and I have wonderful results with MC. So you have to ask those people, actually those who are working in the same, with the same technology in your...
It's all about evidence. It's all about evidence. If it's worked, I think we want publications.
Can I raise my hand?
Yes,
saying something. So, okay, Pranay, you want to take it first?
Thank you
I'll
so
wait.
much. I would want to make a comment that with any of these technologies, I think this is the right point to make this comment. Let none of these technologies constrict our vision so much that all that we have to offer to a patient who has failed a cycle anywhere else is this particular technology that we are adapting in our lab. Whether it be time lapse, whether it be microfluidics, whether it be MZ, I'm pretty sure not all of them. I know patients who come up with morphology reports of 2% normal, and I know this is coming from this particular center. I know patients who come from a particular specialist who for all failed cases recommends ERA, whether it is due to failed blastocyst conversion, poor quality embryos or whatever. So let us not get carried away and look all our patients with a constricted telescope.
Absolutely.
Yep.
Yeah.
And then I think we should
he's put the name.
No, I think we should move away from this wonderful result. We would like to know what is this wonderful result actually.
Since we do not have evidence, since there's not much of evidence in any of this, is there one of this technology?
To the wonderful results.
Sorry, Keshav. Yes. So I know since we do not have none of these technologies which comes under that wonderful category, let's go on the other side and ask ourselves, is there one of these methods where you want to raise a word of concern and say, probably it's too premature to put it onto our patients on a day-to-day clinical usage? Is any of these options that is put on the screen, PASA, IMSI, PIXI, MAX, microfluidics, or usage of testicular sperm in non-azoospermic individuals?
Please, like when you say TISA for non-azoospermic individuals, I would just raise my hand. Like, you need to be more cautious about this.
Wonderful. So that we put a red mark on that area and say, look, there's still a lot of experimentation research is required before we could even encourage this, because it's probably has potential harmful things on the long-term health and safety of the offspring.
It's just something that it's ICSI all over again. You are using the tech for people that are probably not going to benefit out of it, or if they are going to benefit out of it, you wouldn't know why they are benefiting out of it. So you just keep continuing to do it, and it's so invasive. ICSI is not half as invasive as surgical extraction of sperm. And I know there is a lot of evidence out there that says that DNA fragmentation is extremely low, euploidy is good. There is a deluge of research out there. But research aside, let researchers do their research. But research aside, you need to really think about what you're putting the couple through. And there are so many techniques to reduce DNA fragmentation. I would like to give a shout out to one area of research that is ongoing right now in this, which is not on the screen at all. So that's rheotaxic swim up, which is basically creating a ICSI dish where swim up naturally occurs by directly from the semen sample. And it is the most non-invasive project that's going on right now. And we are actually looking for centers that could come in and do that kind of pilot studies with us. But yes, please don't start doing TESA for everyone.
So it's sad for me that, at the end of this slide, I still do not have a concrete answer to take. But I think for all the audience who are hearing out there, I think a lot of precaution, a lot of research and carefulness is needed. I think there isn't any magical step that we could just go and put it across to our patient and say, bingo, this is going to change the whole scenario. So coming to the oocyte, again, where do you want to raise a red flag and say, no, we're going to put a foot down. This is something that really needs a lot of validation of the options put across, or there's something that I've missed out when we're talking in specific to the oocyte or the ovary?
All three of them.
Yeah, everything on the screen.
Yeah.
IVM, spindle transfer, and the oocyte activation.
100%.
Have to be very careful.
So I think though it's new, I think we're still waiting for a lot of clinical validation.
Yeah.
So coming to the media segment, I think, again, nothing is specific to the company of Origio, but, adding GM-CSF for a particular growth factor, they're talking about putting insulin-like growth factors, interleukins, epidermal growth factors, or antioxidants to that matter. Are we really doing it right?
So it's like experimenting. It's like you are taking these embryos as guinea pigs, because at the moment itself, like obviously this media has been tested quite well in animal models, but I'm still not happy with the human research which has happened. And whenever we think of especially culture medias, like Gaurav was mentioning earlier as well, this has a direct effect on the offspring. This has a direct effect on the epigenetics. This can have so many implications later down in the life. And that is where I would want to be very careful of what
the embryo is actually metabolizing, getting exposed, and we do not know what's happening with the epigenetics.
We have no clue.
No clue. So definitely a red flag on this segment of development that we've seen.
Yeah.
The results on animal models are good, which is why they are published. Otherwise, we would never hear of the formulation ever again. But The point is when we are actually using for this patient, see, these companies, they are going to give you a mechanism for this, but they're not actually going to tell you what kind of long-term data, because they don't have long-term data. So word of caution on all of this.
So Dr. Pranay, to you the next question down there with the incubators. Probably, it was more to show that we moved on from a box to bench, a bench top to a time lapse. But if I may be right in saying this, that at least 50 to 60% of a lot of clinics in India still would want to go with a box type of incubator. What is your take on this?
I think even if you're starting out with whatever number of cases, it's always a good idea to have 2 incubators and start with a box and a bench top. A starting bench top would cost as much as a box. You obviously need a box to incubate, equilibrate your media. And I don't think it's a,
So is it mandatory? Is there a research which is saying embryo culture has to be bench top and hypoxic and not box type and 20%?
At the end of the slide, you can have the takeaway, a conclusive takeaway that hypoxic conditions 5% O2 is the way to go. Whether it is in terms of blastulation, perfectoderm to ICM ratio, the total cell number of the blastocyst, the implantation of the live birth rates, 5% oxygen is the way to go.
So Gaurav, your take on this? I really don't know. I'm hoping all of you are single embryologists fixed in one center. I don't know if there are any person who've had experience of freelancing. What would be the challenges?
I think most people here do have the experience of freelancing.
I do freelancing.
We are all traveling.
All right. So because, we've always had this challenge and concern. We go to conferences, people say it has to be a bench top with a 5% oxygen. I go back. I've got my own challenges and I can't be doing this. But has research come to a point where it says only a bench top and hypoxic culture?
Yeah, can I say something?
Yes, Saket. Yes, Saket.
Yeah. So the thing is, it is not mandatory that to go for benchtop incubators, because there are some incubators nowadays where you get a box type incubator with hypoxic conditions. Like Binder, they have recently launched the incubator where you can connect with the trigger structure. So that it's not mandatory.
Sanket,
yes.
But doesn't it give the advantage of a low volume?
Low volume in the sense time to recovery.
Yes.
Time to recovery of the that is the only drawback. That is the only drawback, because and also it depends on the cost effectiveness of the center, because all those things has to be taken consideration. Because if you are doing less number of cases, I think it's well and good.
And then no. See, I would like to ask you one thing. So just because you're doing less number of cases, do you charge anything lesser?
To be honest, To be honest, if I say the packages here, really they're very low, but because of the trust.
I think let's move away from that.
Yes, Keshav, one last comment from you.
Yeah, so as far as the batch IVF practice is concerned, I think we have to be very, like most of the IVF units which are doing batch IVF, the transfers usually happen on day 2 or day 3 maximum. Most of the batches would not continue till blastocyst. And that is where the benefit of the bench top, you can't really analyze, because you will still get good quality cleavage embryos, but your blastulation rates definitely increase. Like Pranay was saying, the number of cells, etc. All of these things definitely increase in a benchtop in a trigas incubator, and that's one of the reasons why batches might still work with box incubators, but you need to have good number of box incubators so that you don't compromise on
absolutely. So the point I wanted to stress here to all my audience out there was, this debate will never end. I'm sure there are a set of people who have had wonderful results with their box type incubators. This is where your documentation, timely audits, looking at KPIs, benchmarking should be the way. Because what has worked with me might not be working with Keshav or my other panelists. But at the end, do you have an SOP? Your clinic, does it have a set KPIs? As long as you're actually reaching those KPIs, I think you're doing a wonderful job. So technology cannot always be the same for all of us universally. If that was true, I'm sure we would have had a different picture scenario of ART all across. So coming to the embryo selection, I'm sure again, we can debate the whole day when we're talking about this. Each one of us has had a different experience with all of this, but I'm sure let's have one comment just on the artificial intelligence. I think now everything is moving on to AI. Is this the right direction that we are moving? Would it really add a lot of benefit?
Yes.
I
believe it
I
would.
definitely, yes.
Yeah,
Yes, Keshav and me are on the same page on this. And
Right.
I can see you have taken one picture from my Facebook post. So I definitely believe in it. It's something that's going to help us, not hinder us, and definitely not take jobs away, because I know many people have this question that if AI comes in the lab, are more embryologists, are less embryologists needed? But to be very honest, if you have AI, you still need to do those transfers. You still need people for doing all the auditing of. When you have machines, you need to have a team around it.
So now comes the debate, a short one because we're running out of time. So lab on chip, I think ultimately this entire technology is taking us towards this concept of lab on chip. And there's always been this little concern that man versus machine, and is the embryologist being thrown out of the whole scenario? So Keshav, we start with you, because I think way back in ISAR, this is what was one of your newsletter that you came up with, man versus machine. So do you really think we all are at some point lose our importance in the whole situation and scenario?
Okay, so this actually came out quite a long time ago. I think it was David Gardner's group which worked, like, did tremendous work, and even Dennis Akas's group with David Gardner, who actually created a lot of prototypes with MIT, and they had a lot of tubes going into the chip where like sequential media was coming in and the metabolites were going out and being analyzed, etc., etc. It looks very promising. It looks like this probably is the best way to culture embryos and the best way to achieve pregnancy. But
where's the cost? Really, where's the cost going to really take us?
Exactly.
If it all,
it has had, Krishna, we are not even at that stage. It has had no translation into even practice, even animal models, etc. It has no translation at the moment. So this is a very far off concept at the moment, even though the research has been there for quite a long time. But I still feel that this is quite a long way
promising.
ahead.
So actually, there is one company that is launching a clinical model in 2021, and that is Overture, because we did some work with them. So basically, they have this whole lab on a chip where it's like a biochemical analyzer. You just put in the oocytes in, and you get the blastocysts.
So, Sally, good or bad? This movement in technology taking us towards
I think
lab
it's good. I think it's definitely good for the patients, because it will eventually reduce cost. See, any tech that comes in the market in the beginning, you have this early adopters. If you know that this particular, there is a management terminology, you have early adopters and then you have these laggards. And eventually when it comes to technology adoption, there is the point when it goes down, the cost goes down for the patients. But I think in increasing access to care.
So two goods. So Pranay, what is your word? Good or bad?
I think we have to look at it as a complementary technology. I think it will never, the human touch will never go away. You look at talking about a lab on a chip. I mean, we know of OCCs that would do better with late denudation. We know that 2 spreads of inseminate volume will not perform better, will perform differently based on their rapid progression. There is so much where a human element comes in. And I think if anything at all, the role of a lab manager will multiply. I mean, you would become more sought after coming in of all these
wonderful
devices.
Absolutely. It'll be like you're the pilot, which is driving, or like which is flying the plane on autopilot.
So and the planes just get more advanced. That's
so Gaurav and Sanket, one last comment from your end.
I think I don't like to comment because this is still under experimental phase. So it's
so you're going to wait. You're going to wait.
When you do ICSI, you feel the while puncturing it. You talk about robotic ICSI, but when you do ICSI, you feel the oocyte. You have that yes, because you are injecting it.
So Gaurav, there's still jobs for all of us.
Yeah.
Wonderful.
So talk to them.
Then why are we not even talking about, a new embryologist? If technology we're putting in so much of thought, then when there's a new embryologist, why these rules don't apply to them? Why we just leave them and say it's okay? I'll just let things happen. Let him learn.
I don't know. Doesn't happen with me. I do everything myself.
Well, that's what I said. Most of you are just probably fixed at centers which are doing good, but there are centers, startup centers, where they nowadays there's so many places offering certifications, masters. They finish the course, they come, they oversell themselves in the interviews. No offense to any candidate. They say they can do everything, but we put them on job, and I've had surprises and shocks.
Yeah.
Why rules don't apply to these people?
No, we are not at fault here. It has to be, you have to hold the universities, like they are the ones you should be asking these questions to, because the kind of education that's being offered, like me and you know, like we know the kind of education that comes from Monash, but I can't be sure, and I can't be sure about the candidate's abilities as well. So always whenever I hire an embryologist, I will hire them for like a 3-month, 6-month, like just to have a look whether they actually fit into my laboratory.
So the point is, the embryologist is no different to technology. There has to be that slow step-by-step progress,
validation, etc.
Validation,
Yeah.
and then only introduced into the system.
So we are not giving a new embryologist to do ICSI or even to do denudation, to be very honest with you. We are just going to first see how they do with the assistant procedures. Then we kind of shift them to the andro part of it and let them proceed. We basically want to see how much discipline they really have, because many of them don't have on-job experience. And when they actually have the discipline of working with an andrology,
so I think it's all about striking the right balance.
Yes.
Be it technology, be it the man or the machine. I think a lot of this validation, calibration, looking at the data, and that is when you embrace or adopt. It isn't an overnight change. I'm sure the clinical team or management or ABC can force us, saying that do this, but I think what we got to be doing is striking the right balance. So technology is neither good nor bad. I love this statement. It's what you do with it that makes the difference. So thank you so much for all the panelists for that lovely inputs that you people have given. So since we've allowed for another 2 minutes, quick take-home message from each one of you.
I think I will start with what.
Keshav, you're breaking in between.
Yeah, he's breaking.
Keshav, we couldn't hear you.
How you're going to, like, what kind of patients that are coming into your laboratory, whether it's actually solving a problem, etc. And keep an open mind. Obviously, you might be in for a surprise with certain technologies as well. So keep an open mind about these technologies and just try and do your best for your patient.
Wonderful. Thank you, Keshav. So Sanket, would you want to put in some closing remarks?
Yes, sir. The primary aim of any IF lab will be to maximize the take-home baby rate. So, and healthy baby also. So obviously, if technology is helping, why not?
Wonderful. Pranay, what's your take for all of us?
I think I would want to reiterate evidence and don't do to others what you wouldn't want to do to be done to you. Let not your patients be your guinea pigs. Have enough evidence for data for the technology to be implemented upon them with adequate counseling. So let's not jump upon a new technology.
Thank you so much. Thank you, Sayali.
So my takeaway would be, before you're adopting any new technology, you need to have your basic audits in place and your basic KPIs in place, and your success rates for the basic procedures need to be A grade, and then you can move into the newer technology. You need to take it step by step. It's long-term success, not short-term. So don't use any technology for your shortcomings in other procedures.
Wonderful. Gaurav.
My point will be look at the problems, whatever you have in your lab, talk to your friends, look at the evidence, and look at the cost, whether your center can afford it or not.
I like it when you say talk to your friends. I so like that point that, discuss it. I think that this is, let's take the advantage of the technology. there's nothing wrong in sharing your this. I know there's so many times where I've heard, oh, I've got 100% results. And I'm like, oh, please tell me, I really want to learn it. Let's be
No,
open.
I think, Krishna, I think the best thing is not to go by anecdotal,
Ma'am, that is where I'm coming to you.
Yeah,
I think all of us, I think right from Keshav, Sanket, Pranay, Sayali, I'm sure you've put in wonderful inputs there. But Sujata ma'am, I think you come from that generation where you've seen what it means to physically run the lab with semi-automation. And now you're running a huge set of chain of clinic where you're actually seeing a lot of automation. There wouldn't be a better person to actually balance and give us the final this for this particular forum that we've done today of newer technology. We would really want to get those little 2 words from your experience, ma'am, today.
No, I believe in 2 things. 1, do no harm. 2, our patients deserve the best. So I think I would like to strike a balance between these 2.
Wonderful.
And if there is something which is going to, like reduce errors with, multiple people working at multiple levels, I would definitely, take the help of technology. No doubt in that.
Wonderful, ma'am. That was a wonderful way of actually putting across all your 20, 30 years of experience into 2 little statements. So inspiring and so nice. Wonderful weekend to discuss this. So at the end, I would take this opportunity to thank IRA, embART, and the entire committee, which have put in a lot of hard work to give us the best panel and this opportunity to run this. I'm hoping audience have got the right thing. Thank you everybody for the time and this evening and this opportunity.
Thank you so much.
Thank you for the invitation. It was lovely.
I enjoyed seeing all of you.
Thank you. Bye.
Thank you so much. Thank you, Dr. Sujata ma'am, Dr. Krishna, for a wonderful moderation of the session. And we would like to move on to our next session. And thank you all dear panelists for putting up the controversies as well as enlightening us with all the different views that we had. We would like to carry forward the next session. Dr. Rahul Singh? Yeah, Dr.
The next, the next speaker, the next moderator for the panel will be Dr. Rahul Sen. And Dr. Rahul Sen is currently working as a chief embryologist at Vasundhara and Jodhpur, and he's EC member for ACE and EC member for Rajasthan chapter as well as IFS Rajasthan chapter. And he's been member of ISAR IFS AS Industry, and is a very good human being and very good dear friend of mine. Over to you, Dr. Rahul.
Yeah, thank you, Sanket, for that nice introduction. Am I audible and my slide is visible?
Yes, you're audible. Okay. And your slide is visible.
Okay, thank you. So, thank you for that wonderful panel, Dr. Krishna and Dr. Sujata. So we are moving with the next session, and myself, I welcome my fellow panelists, Dr. Harsha Badarka. She is a director IVF lab, chief embryologist at Akanksha Hospital and Research Institute, Anand, Gujarat. And next one to follow is Dr. Akash Agarwal. He is Scientific Director, Hegde Fertility, Hyderabad. And for the next couple of minutes, we'll be together discussing upon when to opt for extended culture. So, as it is unanimously known that the process of laboratory aspects of ART that begins from the retrieval of gametes, the fertilization, and eventually ends up at the embryos transferred back into the uterus. And in between that, there are the various techniques and processes involved which aids in the maximizing chances of success. And one of the technique is the extended culture, which is the culturing embryos till the blastocyst stage. So there are the advancements in culture media, techniques of fertilization, gamete selection, and embryo culture, which has increased the prospects of extended culture. So now the actual question arises, what and why we need this technology. So what is the actual need of getting embryos till day 5? I would like my fellow panelists to comment on this. To first to follow is Dr. Harsha. Dr. Harsha, can you hear me?
Yes. First of all, very good evening to all of you, and thank you for inviting me as a faculty member to organize this. So why the need of blastocyst culture? As we know, embryo selection, way to is better in blastocyst culture. You can select better competent embryos. You can select single embryo for SCP. To synchronize uterine embryo communication. These are the factors which allow the blastocyst culture in our lab.
To add on with these steps, Dr. Akash, would you like to add something?
It will, what I would like to add on is we can decrease the multiple pregnancy rates. We can go in more towards a single embryo transfers because we are increasing the implantation rates, and to help us in this will be embryo biopsy, which is obviously proven to be more robust at day 5 stage. So it could be as a part of your day 5 embryo biopsy stage also, a part of that. And obviously I would like the most important thing is increase the implantation rates. And as has been told by Dr. Harsha, increase the implantation rates. I think those should be the most important things. And definitely to reduce the multiple pregnancies. You know, there's nothing worse than you have a patient like who has been struggling for 5 years or 10 years, you transfer 2 embryos and, you know, they abort at early 24 weeks or 21 weeks. That's the worst case scenario you would like to see.
Yes. And eventually the literature is flooded with the information that yes, blastocysts do really increases the efficiency in terms of success or the outcomes in ART. So with this much of positivity over blastocyst, that everything should be well and worse and fine with the blastocyst outcomes, do you think there are any setbacks of getting an embryos up to blastocyst stage?
See, drawbacks are you need to culture 2 days more in the lab. So your lab should be set as per that. Secondly, you will end up with less number of embryos comparatively cleavage stage embryos. Sometimes you are ending with nothing. So these are the drawbacks of blastocyst culture.
Yeah. To add on with this thing, Dr. Akash.
Yeah, most important thing would be not to have an embryo for transfer, like rightly said. And, you know, your lab has to be really good to be able to take it to day 5. You know, the conditions what are required by a day 3 embryos, they are not as exacting as they are for a day 5 embryo. So not to say that you should have a suboptimal lab conditions, but, you know, a standalone center, which is probably there in the periphery, we should not get carried away by trying to mimic others. And we should see what works best for us and try to culture once we achieve our KPIs with respect to our cleavage stage embryos. So once we do that, then only. And obviously it does not work for someone who does in batches or someone who is an on-call embryologist. You know, they will not be so much willing for it. So you need to have an in-house. You know, you say on day 5 in the morning, you'll be seeing, and what you see is the early blast, or say it's something like a 3A stage. What do you do if it's an on-call embryologist? So you need to keep those practical things in mind.
We'll get back to these points soon because they are coming in the upcoming slides. So as it is mentioned that yes, there are the positivities and the negativities that out there with the blastocyst. So I think there must be some criteria or some of the indications which has to be taken care while taking any couple or taking any embryos up to the blastocyst stage. So what can you say about it? First to go with Dr. Akash, can you highlight on this?
When we go in for a blastocyst culture, the first thing that we can do is right from the history of the patient. what's the age of the patient. If it's a young couple, we are expecting a good prognosis and they don't have any bad prognostic factors, anything like no PCOS or endometriosis. These kind of patients, especially below 35 years of age, they'll try to get more number of eggs and the quality will also be good. So for me, the first criteria would be the age of the patient. What are the semen parameters if they are normal? And next would be the number of embryos. It's very important that we have, I would be more comfortable if I have something in the range of 4 grade 1 or grade A on a day 3 for them to be carrying over forward. We have cases where we have seen lesser number. In that case, we just speak with the patient and tell that they have a 5 to 10% chance of not having a transfer at all. We always try to include the patient in that discussion. One more thing I would like to do is it's an individualized decision. It's not one thing works for all. Yeah. So you have a patient who is 40 years old. Now we are having a patient who is 40 years old. She gets only 3 embryos. They are not of that good quality. So what your choices would be? So I would rather prefer a cleavage transfer for such a patient rather than take them.
Actually, so the same thing goes on with Dr. Harsha. Dr. Harsha, what are the embryological parameters, as Dr. Akash has clearly mentioned about the age and the other parameters from the clinical side of the patient? According to the embryology parameters, what do you think those might be the conditions can affect or can be a selective points when we can proceed with the blastocyst transfer of the cultures?
Your culture conditions, your lab conditions, embryo or blastocyst scoring should be included in the selection method. Good quality and quantity and quality embryo is great for blastocyst culture. All these criteria should be included in blastocyst culture technique.
Yes. Nicely presented by Dr. Harsha that yes, embryo scoring or the blastocyst scoring should be a key point at that time. And we have already seen with the literature that earlier times we used to get the blastocyst scoring on the basis of the fact that which was devised by Dokras et al. Way back in 1993, and it was solely devised on the expansion of the blastocyst. And now we have assumed the fact that the sufficient number of blastocysts are there available, so there is an actual need of a scoring system so that we can select a best embryo or the blastocyst for transfer. And we can see in the in vitro culture conditions that blastocysts of different qualities can be seen per patient, per embryo, and thus the requirement of or establishing a good scoring system has to be there. So following with the scoring system that was the most widely accepted, which was devised by Gardner and Schoolcraft in 1999, which was devised on the basis of the fact that how's the expansion according to the expansion of the blastocyst, the type of inner cell mass, the compaction of or the trophectodermal layers, trophectodermal lining. So coming back to both of you, that on the day of transfer, under the microscopic situation, we see something like this. So now I would like to know from both of you that which one will be your best selected blastocyst for transfer?
4AA.
Yeah. What will be, according to you, the definition of a top quality blastocyst, and how is that relatable with the transfer, means the blastocyst transfer?
You asking me, Rahul?
Yeah, Dr. Harsha.
So top quality blastocyst means very well expanded blastocyst with compact ICM and very good trophectoderm cells. We can say perfect 4A blastocyst together.
Yes, Dr. Akash, you have to.
First, I would like to know if I have some time to wait. I would rather wait till the evening. If this is on the day of the morning, I would rather want the 4AA to start hatching. That's the whole point of having assisted hatching to take place. So I would rather wait for some time and see if the 4A starts to hatch first. Then that would be my first choice if I can wait. If no, 5BB, definitely embryo which has started to hatch first would be a better choice. And the rate of progression is also an important criteria. Agreed, ICM and TE are there, but even the rate of progression, hatching definitely has to take place before there has to be implantation. That would be the first thing. And what I would like to add is regarding top quality blastocyst, I would say something about 3BB or more. Anything which is 3BB or better would be a top quality blastocyst because they give almost equal implantation rates. I would not say they are equal. Say 3BB and 5BB, obviously the 5BB is going to give you a better rate. But still they are comparable. So a top quality blast is anything which is 3BB plus on say day 5, 116 hours.
Yes, as clearly mentioned by Dr. Akash that yes, 3BB will be a cutoff according to him. So Dr. Harsha, do you agree with the fact that the 3BB is the least, or what we can expect on day 5, that at least 3BB should be there, or below that if it is seen you can get it transfer done?
See, 3BB is the cutoff, but you cannot thrown out or discard 3CC also. Sometimes in favorable condition you can get results. If you do not have any option, you can transfer this 3CC also. So it depends on the options. Obviously 3BB is cut off. Up to 3BB you can get good results.
So as rightly mentioned by both the panelists that yes, the good quality, the top quality blastocyst has eventually the most positive outcomes in ART. Not only the blastocyst scoring, but yes, as clearly mentioned by Dr. Harsha in the previous slides also, that the culture conditions are the key factor, key points where blastocyst growth can be proceeded. Like we see a lot of different points where we sometimes puzzled up that whether these conditions can be there or can promote or demotivate our blastocyst development, like whether culturing embryos in a group or in a single with the interruption or non-interruption into a humidified incubator or a non-humidified incubator. So there are a lot of things. So according to you, what set of conditions do you prefer while getting a good blastocyst cultured? Yeah, Dr. Harsha, yeah, you can begin the first.
See, in between mass culture and droplet culture, I prefer droplet culture.
Droplet culture with the minimum embryo density of
single droplet media and single embryo.
Okay.
So obviously mass culture has few benefits. You can have some embryotrophic factors inside it, but also some toxic substance also increases. So I prefer droplet culture. Interrupted versus uninterrupted culture, I am using single step media, but still I'm changing media on day 3. So uninterrupted culture I prefer. I'm using I'm using humidified incubator, and I have an embryo scope, so it is non-humidified. So I have both incubator and having good comparable result in both the incubators. So there is nothing difference between humidified and non-humidified incubators. Renewal of culture media at cleavage stage, as I said, I am using single step media, but I'm changing media on day 3. Oxygen tension, I'm using 5% oxygen, so I do not have any experience related to oxygen tension in incubators.
Anything you would like to add up into this, Dr. Akash?
Well, I would like to slightly differ for a couple of points, I guess. Interrupted versus non-interrupted, and that's a no-brainer. Like obviously, if we have the luxury of having something like a time lapse, then yes, that would be the obvious choice. But right now, I'm not having that luxury. But there's no doubt that we want to minimize the exposure. As far as humidified, I have experience with both the humidified in terms of a benchtop and non-humidified. The literature says the humidified is definitely good. Personally, what I have seen, probably the number of cases, I have not seen that much of a difference with the humidified and non-humidified as well. Next is a sequential versus single step. Somehow I'm a big fan of a sequential culture. I mean, you can have an entire webinar about it, which is the best one. There are people who do single step a lot,
but yes, there are many who are favoring with the single
I
step.
know that single step, but could be something to do with the lab conditions that I work with, or don't know. But see, it doesn't make a point if you are going to change it on a day tree, like Dr. Harsha has said. If you are going to change it on a day tree, then anyways, it's not uninterrupted. And I would prefer a droplet culture. Your paracrine factors, and my personal experiences is it's definitely much worthwhile to have a group culture of at least something 2 or 3 embryos, and you reduce the volume. Don't go in for a 0.5 ml. You can make it a droplet, a smaller volume, something of 100 microliters or 200 microliters, and put 3 to 4 embryos of the blastocyst. So you get the benefit of both the worlds, where you are having the benefit of paracrine factors, as well as you have the benefit of not too much of accumulation if you are worried about that. And oxygen tension, again, it's a 5% low oxygen, hence we have all our benchtop incubators.
Yes. So it is already mentioned in our previous webinar also that yes, oxygen tension at around low oxygen levels tends to give a good blastulation or the good outcome in ART. So moving up with the next set of situations.
If I add something, Dr. Rahul? There was a question in the panel also. Is it a good thing to culture till day 3 cleavage stage embryo and then go ahead for in a normal box type incubator, let's say the normal oxygen levels atmospheric, and then go in for a day 5? There are studies which say that rather the high oxygen level is going to impact the embryos more at the earlier stage, the earlier they are. So it's the cleavage stage embryo actually which gets the maximum impacted by the concentration of the oxygen level. So I would definitely prefer it right from day 0.
Okay, so it's from day 0 you'll be sticking with the ultra low oxygen conditions, or means what will be your
I don't have that much experience with ultra low.
Okay.
Till 5%, yes.
Okay, 5% will be your
5%, 5.5.
So now we have taken care of all the parameters, all the factors, and we have taken care of the culture conditions. And on day 3, we are having a situation like there are the good quality embryos, but there is a factor of limitation with the number. And on to the other side, we are having a means what we can say average quality of cleavage stage embryos, but there is no limitations with the number. So now the question is still, can we proceed with the blastocyst transfer in both these situations, or would you deny, or what will be your preference in these cases?
Good quality and good quality and cleavage stage embryos. Suppose you have 2 or 3 A grade embryos.
Cleavage stage embryos, yeah.
Cleavage stage embryos. So I will prefer day 3 transfer, cleavage stage transfer rather than blastocyst transfer, because I think you would not have any selection criteria after at least 2 blastocysts. So already you are going to transfer. So in low number of and good quality embryo, obviously I'll go for day 3 transfer. For second question is average quality and large number of embryos.
And good, yeah, the number is not the limitation here.
So in this case scenario, I will prefer, because embryo quality is not good, I will prefer day 3 transfer, 2 or 3 embryo on day 3, and then we'll allow raised embryo to go for blastocyst stage, and raised blastocyst you can freeze. Because sometimes I have experience, I transfer in 3 very bad D grade embryo in fertility preservation patient where husband had a testicular cancer, and we had only 3 D grade fragmented embryo. We transferred it and we got results, because wife's age is around 28 years and everything is normal uterine side. So, and we got very good, we got live birth with baby girl. So this happens sometimes. If C or D grade embryo gets favorable conditions, they can implant. So I will prefer day 3 in this case also, and rest you can go for blastocyst.
That's perfect. Anything you would like to add on with this, Dr. Akash?
Yeah, scene 2, absolutely spot on. Like no difference in the answer, I would say. That's word to word what I would have liked to say. Because there are studies which say that if you have a bad quality embryo, the implantation rates are better, almost twice if you do a transfer on a day 3, almost twice. With respect to scene 1, I would still agree that it would be a cleavage stage. The only time when I would want to go it for a blastocyst is, say the patient has failed previous 2 or 3 times with the past history in mind, then I would like to know whether the embryo is progressing to day 5 at all. That is one thing which we can answer. Obviously, it is with the patient's consent, counseling, proper counseling. But we have done cases like that, and you don't get a blastocyst. It's more easier to counsel the patient towards the other choices what they have.
Yes.
So I would prefer, if it's a early one, it's a first 1 or 2 cycles, yes, cleavage stage. But if they have failed implantation, then I would like to take them for a blastocyst even in the first case.
So in this particular situation where you see no blastulation or still compaction on day 5, whether would you like to proceed it further on day 6 or day 7? Any experiences of extending the blastocyst culture till day 6 or day 7 with the average quality embryos and getting those culture till day 7 or day 6?
That's for me.
I do not have.
Dr. Harsha, Dr. Harsha, you can.
I do not have experience of day 7, but yes, I have recently communicated a paper in which what I did, day 5 compacted embryo, which are not expanded on day 5. We freeze them, and then we thought at the time of, as most of us, we are doing the frozen thawed cycle now. So we freeze day 5 merged embryo, and at the time of transfer, what we do, we thawed them before 1 day, day 4, and then let them grow up to post-extended culture. And then in this study, we already got a very good results, and we are publishing a study in 6 months. So I do not have experience of day 7. Thus, yes, day 6 you can go. There are literature which shows day 7 embryos are about 5 to 8% day 7 embryos are usable. So you can, they may be non-enucleoid embryo, and you can transfer that day 7 embryo, but after freezing, this thaw cycle, you can use this day 7 embryos. Rather than discarding this embryo, you can use it.
Yeah, as rightly mentioned by Dr. Harsha, that data from literature has suggested, yes, on day 7, there may be embryos with euploidy, and maybe embryos available for transfer. Maybe we can get a positive outcome, but still there is a controversy behind it. And it may adds up with the extra usable forms of embryo. And with the advent of cryopreservation, as rightly depicted by Dr. Harsha, that freezing can give a good option to at least preserve those embryos and do the transfer later. So the cryopreservation with blastocyst represents a unique challenge due to its multi-structural or multicellular features of the stage and the development, with the presence of blastocele, and the blastocele cavity is fully with the water content in it. So according to you, what can be the factors that can affect a blastocyst cryopreservation? Yeah, Dr. Harsha, yes.
As you said, blastocele cavity includes the formation of ice crystals. But I think nowadays all embryologists are well trained. So in my clinic, we are getting good results, good survival rate in blastocysts, around 95%. So I don't think there is any difficulty in blastocyst freezing. There are few advances that you can remove blastocyst fluid by laser hatching or by aspirating fluid, and you can do freezing method. There are few tips for freezing, but blastocyst freezing nowadays is easy for us.
Yes. Anything, Dr. Akash, you would like to add up with the optimizing blastocyst cryopreservation? Because now it's high time that we have got the blastocysts, but yes, they are the surplus blastocysts available. We can do the cryopreservation.
Yeah. One thing would be like you have put in your slide, the best thing is, most important thing would be the blastocyst quality. What goes in comes out.
Yeah.
Right. So definitely that would be the most important criteria. Your freezing rate, warming rate, operator dependent rate. Yes, your learning curve. What I would say rather that the blastocyst, it gives you a much more cushion than a cleavage stage embryo. The later you vitrify an embryo, it's going to be better. The results are going to be better. You lose a couple of cells also, it's going to help you. So I would be rather more comfortable freezing a blastocyst. Yes, we have hit almost levels of 95. I would say almost 98 or 99. And it more is usually something, to do with the blastocyst that has happened during freezing. Sometimes we have that which have not shrunken properly. So we do make a note, anything deviation from the SOP that this has happened. And usually these are the blastocysts which we tend to lose them until unless there is any fault from the operator side. I mean, that is altogether different story.
Yes. So as rightly mentioned by Dr. Harsha, that there are the various techniques available by which we can actually shrink the blast and we can reduce the water content so that a high quality or a good outcome can be optimized with the blastocyst cryopreservation. So now moving up with the another set of condition that we a completely new setup, a completely new lab is there, and now they have done with the cleavage stage embryo transfer. They are looking for blastocyst transfer. Means when should they begin, or any other points they should be like taking care of before commencing a blastocyst culture in a newly constructed or a newly started lab? Means
So,
yes, doctor. Yes, yeah, Dr. Harsha. Yes, you can.
See, we just moved before 3-4 years to new setup. So here I will advise that you need to do simultaneously both the setup run for at least 15 days. What I did, the first embryo I cultured in new lab is embryo donation. We use donor egg with donor sperm. We let them grow up to the blastocyst stage, and then good blastocyst we transferred into patients, embryo donated patients. And we got first case results. So I advise that first check your culture condition after setting up your lab as per quality assurance and quality control. You just culture one set of donated embryos, and then you can go for patients. So it will better for patients also. So I advise the first case of embryo donation in new setup.
Yes, exactly. As rightly mentioned by Dr. Harsha, that embryo donation or checking with the quality of blastocyst earlier before getting into the patients directly, it can be a good option. So Dr. Akash, anything you would like to add up in this with the blastocyst?
One thing, that's a very good point, I would say. You can always go ahead with the embryo donation. These are the patients, not that they are any less worthy, but you always have a chance of giving them another shot at it. Okay. And your KPIs on day 2 and day 3 are performing well. And then obviously in a good prognosis patient, we would like to split it and say, even if it's a freeze all cycle, we individualize. What we do, we freeze, say you have 6 good quality embryos, grade A's on day 3. So we would freeze a set, one set of it on that, or either transfer or freeze, and you can take the remaining of them for day 5. So, you're not entirely compromising the cycle. I would say it's not exactly an experiment because you're having 6 good quality on an average, I'm saying. That is a number I would be more comfortable with. And I still remember the first day when I had got it, the good blastocyst. I can never forget the first patient I had got. It was luckily a 4AA. And that always gives you the kicks, to go ahead with that. Nothing better than that.
Yes, and there has been a lot discussed about the KPI. So what has been the role of KPI in means how these KPI revolve around the blastocyst development with the cryopreservation or with the blastocyst that is getting produced in the lab. So any thoughts of your thoughts with the KPI? Dr. Harsha, you can begin with.
See, blastocyst development rate is around 40 to 50% of your patients, but it is very depending on individual criteria of individual indications of patients. Sometimes in male factor, severe OAT patients, you can get very less number of blastocysts. So it is, the benchmark is that you have already rated around 40 to 50%. Competency is around 60%, because if your lab is generating aneuploid embryo, more aneuploid embryo, it is you need to check the quality control of your lab. So it should be more than 60% competence embryo blastocyst development in your lab. And for freezing, obviously around 90-95% survival rate should be there.
Yes, yes.
May I add something?
Yes,
I think
doctor.
that's just a typo error. It's rather the competence would be at 40 and benchmark would be at 60.
That is for the blastocyst development rate.
Yeah, blastocyst development rate. It would be the competence, the minimum level that you are supposed to achieve. That would be at 40. And your benchmark would be like what you aspire for. That would be at 60. I think that's just a typing mistake. And we usually see regularly blastocyst rates in average of 50 to 60%, I would say. Until unless they are bad prognosis patients where we get less than 20 or 10 might be, hardly 20, something like that.
So now with the final conclusion we are having, it's like, can we proceed with the blastocyst cultures or the blastocyst transfer for all, or there should be a selective approach? The patient selection should be there, or the embryo selection should be there, getting embryos done with the blastocyst?
I'm one for an individualized transfer. It's like never, I never believe that one set applies for all. I'm a strong proposition for it. I would be like, go ahead with the blastocyst transfer wherever possible.
Yes. Yeah, Dr.
Yeah, I will
final
go with
comments?
Akash.
Yeah.
Yeah, I will go with Akash.
Yeah, just to conclude this session, yes, blastocyst gives a good outcome. There are the chances of having increased live birth rate as compared with the cleavage stage embryos. They gives us another optimization with the elective single embryo transfer, and there must be a reliable grading criteria for cleavage stage and the blastocyst stage embryos. Yes, at the end, on the day 5, this picture is really a eye-touchy picture, and everyone would love to see these kind of embryos growing into the culture conditions and getting these embryos transferred, getting a healthy outcome out of that. So thank you, my co-panelist. It was a nice evening to have you with us, and I'll proceed with the further agenda
Thank
of
Dr.
today's
Rahul
meeting.
for a wonderful session, and thank you, Dr. Harsha.
Thank you, Dr. Rahul. Thank you.
Thank you. So I think Dr. Sanket will take over the session.
Yeah. Thank you all for the wonderful panel. Can I have the screen, please?
Yeah, I have stopped.
Yeah, here comes the next moderator for the session, Dr. Shrikant Nale sir. So he's a well-known person in Indian embryology fraternity with having more than two decades of experience. Over to you, Shrikant sir.
Yeah, hi, good evening, everybody. Before not wasting the time, we are already half an hour late. I'll just fastly introduce. No need to introduce. In fact, Dr. Shubhangi Kangal has been working in this field for more than 2 decades now. She has done a PhD in 2009. Had been undergone training at Italy, Singapore, and she has experience at different centers, and she's working for more than 20 years now. And at the basics to art of making the dish. So that where we start with. Very important, like key factors, what we culture, where we culture the embryos. So Shubhangi, to you.
Yeah, thank you very much, Shrikant. And thank you very much, IHERA, for giving me this opportunity to talk or give a lecture on dish preparation. So this is like the very, very basic first and the foremost step that any embryologist would be performing when he enters ART lab. So we all know about this. We are all working as embryologists, maybe junior, senior, for over years or even few months. We all of us know what is a dish for embryology, for embryo growth, for assisted reproductive technology. So then what is the need? Why do we need to know more about this? So my dear friends, I'd like to tell you that this is the first step to the creation of life in an artificial setup or in a laboratory setup. So we as embryologists are creating a stage for the most important performance of you where I mean is us as embryologists to begin with. So if the stage is set right, only then your journey in this field of embryology would be a wonderful and an interesting one. So basics of dish preparation goes hand in hand with the type of embryo culture system that you're going to be using. Why? Because the embryos or the gametes are going to be resting there in your dish, which you are going to be making a day before or on the day of your ART, various ART procedures. So what does a dish preparation require? It requires the media, it requires the gas phase, it requires the medium of overlay and the culture vessel. Finally, we, the embryologists, are going to take charge of these 4 parameters while we are making a dish for the embryo culture. The incubation chamber or the incubator and the ambient air quality also are important, but they are beyond the scope of my lecture today. So coming to optimal way of dish preparation. Why, how good or how bad or how optimally rather we should prepare a dish? So the answer to this is we need to understand the basics. We need to understand the effects of osmolality and temperature, the effects of these 2 parameters on the embryo development. If we are sure of these 2 temperatures, how are they going to affect your embryos, which are going to be sitting in the dish, in the incubator for maybe a day, 2 days, 3 days, or up to 5 days? Then you need to know this. If you are aware, if your basics of these 2 parameters is clear, and then you are going to get very good embryos.
Hello, Shubham. Sorry to interrupt. Is there a screen view? Can you see the screen? Are you sharing the screen?
Oh, am I not sharing the screen yet?
No, I can't see. At least anybody else can see?
How do I share it then?
The bottom of
we are not able to see your screen.
Yeah.
Okay, one sec. I'll just...
Bottom of that window, there would be a share screen, a green color, this thing.
No. My technical expert is helping me out. I'm sorry.
At the bottom of the Zoom window, there are 3-4 options: question and answers, chat,
share screen. Yeah, I got it.
Share screen.
Yeah.
A green one.
Share the complete screen, ma'am.
Yes. I'm doing it. Okay. Is the screen seen now?
Yeah.
Just
Make
start
it a full screen.
Make it to slideshow, ma'am.
Make it slideshow. Make it slideshow. Make it slideshow.
Or you can press F5.
Yeah. It's now there. Yeah. Thank you.
Thank you very much, Shrikant.
Thank you.
And otherwise, I would have just gone on and on talking about my dish preparation.
I thought it was an introduction.
Thank you so much. Shrikant, it's a pleasure that you are moderating my session. We both go a very long way in this field of embryology. Remember the days when we used to, work on different...
Very much.
Yeah.
As time was limit, we were already running late, so I didn't.
Okay. So, like I was saying, this is the first step to creation of life in a laboratory setup. You are creating the stage for the most important performance of your life as an embryologist. So these are the important sorry. The media, the gas phase, the type of medium overlay, and the culture vessel are the essentials of making a dish. And we as embryologists, we always observe this, but we seldom ponder on this, that why is it so important? Why are we going to do this? What is the significance? So as I was mentioning before, we need to know the basics like the effect of osmolality and temperature on the embryo development. So as such, if you see, dish preparation is very simple and easy. It requires no prominent skills, but alertness definitely. So what are the steps of preparing a dish? A dish, it serves as a base for the culture media or the buffering media, which will be harboring the embryos, the oocytes, the sperms, the zygotes, and all that is on a daily basis that we are seeing in our embryology laboratory. The overlay will be mostly the culture oil or the paraffin oil. Why? Because it prevents the evaporation, maintains the homeostasis, prevents the pollutions by the volatile organic compounds, and thus sustains the sterility in the dish that the embryos are growing it. Now, in the ICSI dish, PVP is important to immobilize the sperm, and we use hyalase or hyaluronidase for denudation before ICSI. Now, why is osmolality and temperature important? So I was just going through a lot of papers and I found this very interesting paper, and it is by Jason Swan, and he measured osmolarity using different parameters. So what was done was he made dishes using a standard method for dish preparation. That is, you make droplets and overlay with oil, or by using the wash method to prepare droplets, wherein the droplets are made and then they are refreshed under oil by the culture media. Now, he also observed that whether there was any difference in the osmolarity when these dishes were made at 37 degrees or at the room temperature. He also made use of the laminar airflow. So the laminar airflow was switched on and the laminar airflow was switched off. And he also varied the droplet sizes. So he made dishes using 10 microliter, 20 microliter, or 40 microliter droplets. Now, what was observed, he did all these experiments and also measured the osmolarity 5 minutes after the dish preparation and 24 hours after the dish preparation. What basically he observed was that the osmolarity did not change. There were no differences in the results between 5 minutes and 24 hours, sorry. But what was observed was the droplet type. So the standard droplets, if you see here, the standard droplets, the osmolarity was maintained. The standard droplets, the osmolarity was increased, and the wash droplets droplets made by the wash method performed better. If you see the volume, then definitely in a 10 microliter droplet, the osmolarity was very high, and as compared to 40 microliter, where the osmolarity increase was less. Now, when the airflow was switched on in the laminar hood and whether it was switched off, there was not much of a significant difference between the two. But definitely there was a very statistically significant difference seen when the droplets were made at 37 degrees rather than at 23 degrees. So what was observed that at 37 degrees, the osmolality of the media definitely changes. Now, we need to know that embryos will develop best, or they will have optimal development only when the osmolarity is between 270 to 290 MOSM per kg. Now, if you look at a graph here, you will realize that there were different osmolarity solutions that were used: 270, 290, 310, and 330. Now, at 330 degree osmolarity, immediately, even at the cleavage stage embryos, you would see that there was development was hampered even at the cleavage stage. So if you go ahead and see at 30 hours, again at 330, the development was very bad. It didn't develop as well as it when the osmolarity was between 270 and 290. So subsequently, the early blast and the expanded blast and the hatching blastocysts performed best when the osmolarity was maintained between 270 to 290 MOSMs. Now, there was one journal in which I read an article which said that whether you have to make the dish, you have to prepare the dish immediately before the procedure, or you could make the dish 2 days prior to the ovum pickup procedure, and whether there is any significant difference between the two. Now, this 2-year study was performed where 76 cases were studied and 152 were matched controls were in. So it was a huge number of patients that were studied, and the main embryological parameters was cumulative pregnancy rates and the main embryological parameters with respect to the blastocyst formation and the pregnancies. It was similar in both the types of dishes. The dishes which were made 20 hours before the ovum pickup, here they had included a Sunday. So actually it was 48 hours prior to the ovum pickup, or generally if you are making it 6 to 7 hours before the embryo ovum pickup. Now, there was also a study wherein it was found whether there was a significant difference when the embryos were cultured in the 4-well dishes and in the center well dishes or the 60 mm dishes using micro droplets. What was interesting that the fertilization rates were lower when the embryos were made cultured in the 4-well dishes, and also there were more aneuploid embryos in the 4-well dishes. But there was no significant differences on the embryo stages on the consecutive days. So this was a very confusing conclusion, I would say. Now, if you are going to be culturing embryos in an open culture system, wherein you use a center well and you put some media in the moat, versus closed system wherein there are droplets overlaid with oil. Now, we need to know that both these types of systems of dish preparation has their own advantages and disadvantages. Open system exposes the oocytes and the sperms to the changes in the environment in which they are growing, mainly the pH and temperature fluctuations. Closed systems are definitely better, but in a droplet, there would be competition for nutrients between thousands of cumulus cells of the oocytes and the thousands of sperms which are present at the same time in one droplet. Now, we are not very sure of effect of metabolites on the oocytes when we are culturing those sperms and oocytes in a single droplet, while specifically doing the culturing for IVF, that is in vitro fertilization. But most commonly used method is droplet culture under oil. Next is whether you are culturing your oocytes in a group in a center well which is covered with oil, or you're culturing your oocytes in a single droplet covered with paraffin oil. Now, there are claims of better embryological outcomes in group culturing because of the paracrine and the endocrine influence of the cumulus cells, which when they are cultured together, help each other to grow. But the single droplet culture definitely gives higher fertilization rates because there is homogeneous sperm distribution in a droplet. Now, this definitely will vary, and it is a topic of debate because every embryologist will be having their own experiences, their own habits, and their own knowledge and their results in their own laboratories. So, but definitely there is a better control over embryo development in a single droplet culture. We need to unify worldwide while this is concerned. In my lab, when we are doing IVF, I use a center well dish with 1 ml of culture media, which is overlaid with around 1.2 ml of oil. And we inseminate around 2 to 3 lakh of sperms per 1 ml. And the oocytes in one center well will be around 4 to 5. When we are doing ICSI, the dishes preparation is by the buffer media, which is most of the times hippies. Now, when we are denuding the oocytes, then I also use the centerwell dish, wherein I will be putting around 40 to 50 microliters of Hylase in 0.5 ml of hippies media. I will also be taking hippies media, which is preheated in the moat, and I would be denuding the eggs for ICSI. ICSI dish preparation is slightly different, wherein a dish needs to be preheated. It has to be prepared inside the laminar airflow. The patterns of the droplets will vary. Again, this will depend on embryologist, how he is comfortable with the way he wants to perform the ICSI. But by and large, if the sperm count and morphology is good, generally one PVP droplet with media droplets is advisable. If the sperm count and morphology is poor, then in order to expose the oocytes for a lesser duration of time, it is better to make 2 PVP droplets to facilitate the sperm preselection in one and creating a reservoir in the second droplet. PVP droplet must be flat so that you could touch the bottom of the dish and immobilization of the sperms is faster and easier. We have to add optimal number of sperms in the sperm droplet to avoid overcrowding. Otherwise, the sperms which are in the interphases on the flat droplet are going to create a confusion and a little disturbance to pick up the best sperm and crushing the tail to immobilize it to further inject for ICSI. Now, most important thing we need to remember is that once you are making a ICSI dish, the droplets are extremely small. They are sort of micro droplets. So important that you see that they are submerged completely under oil. How do you see this? Is by keeping the dish under your stereo zoom microscope and make sure that all the droplets are completely covered under oil. We have to use PVP at room temperature and oil which will always be preheated and pre-gassed at least 24 hours before the ICSI dish preparation. Now, in conclusion, I would like to say that we need to remember that prepare dish one by one, don't be in a hurry. Labeling is most important. Make sure that your labeling is clear and very concise, and you're writing the purpose of the dish. So in our lab, we write S for the scan dish, D for the denuding dish, and C for the culture dish. Witnessing is very important. If you have very high-tech RI witness or wonderful, but if you don't have, make sure that there is an assistant or your senior, whoever is making, witnessing the dishes while you're making the name of the patients and for what you're using the dish. Now, place the droplets in the dish using a variable volume pipette, which is fixed at 40 degrees if you're making droplets for insemination for IVF, and immediately cover it with oil before placing the droplets in the second dish. Now, keep the bottles of media cold at all times, because we know that with the higher temperature, the risk of evaporation is more when the bottle is opened. Now, dish preparation has to be made a part of your QC to facilitate troubleshooting. We have to make sure that you're using certified plastics which are IVF safe in your lab while you're making your dishes. I thank you very much for your patient hearing. And if you want any details about, I just read some few questions on how many sperms do you inseminate for IVF. So if there are any questions, I'm happy to answer. In our lab, we put around 2 to 3 lakh sperms per 1 ml of media of culture, which will have 4 to 5 oocytes. So for example, if your sperm count post-wash is around, say 12 microliters, then I would be putting around 20 microliters of the concentrated sperms in 1 ml of media, which is the culture media in a center well, and it will have 4 oocytes. So if your sperm count is 40, it will be only 5 microliters of sperms, which I would be injecting in 1 ml of culture media. Thank you very much.
Thank you, Shubhangi, for a very good, basic, important thing, topic of dish preparation and media usage.
I'm sorry for the technological glitch that happened, and the slides were not to be seen in the beginning.
I don't see any more questions here, but from my side, I would like to ask you one. What would you prefer doing while doing the dishes? We will allocate the media, heat it into gas in the incubator, and then prepare the dish, or you directly open the bottle, fresh bottle, prepare the dish, and then you keep it for gassing. That is one. And second question is, when you use a single step media till day 3 and day 5 or whatever blastocyst stage we take, do you recommend to change the dish on day 3, like fresh media is to be fresh dish, the embryos has to be transferred, and that fresh media or fresh dish in which the media embryos are to be transferred, are they gassed for 1 day prior, or are the same gassing is done for that?
Yeah, to answer your questions, I use media directly from the fridge, which is cold, and I make the dish 1 day before. So if your oven pickup is at 8 o'clock in the morning, I would prepare the dish probably in the second half of the day, anytime after 2:30, and overlay with oil. So that much time, 16, 17 hours or 18 hours is good enough for gassing, as well as for the temperature to be achieved at 37 degrees. Now, if you're using a single step media for embryo culturing, I do change the dish on day 3, because if it is ICSI or even if it is IVF, the dish is definitely changed on day 3, and fresh media is used. I like to replenish fresh media for the growing embryos.
So you recommend to change the dish, and prior that is heated to be minimum gas to be 12 hours, not more than that. What is the maximum duration of the day or time where a gas media can be used? 4 days or 3 days? What is that? And one more, just added question to that. Like, how frequently do you check the pH? And what gas concentration, like, is it dependent on the media to media? Do we need to this thing, or there is a standard thing like 6.5 or 6, whatever the
No.
is recommended?
Now, the CO2, which is displayed on the incubator, and the pH could vary definitely. These are two different things. So if your pH, I generally prefer my pH between 7. Actually, to be very precise, I like it 7.33. For several years, if it is between 7.33 to 7.35, I think I get good embryos, morphologically good embryos, and I'm happy with that. We generally do pH checks weekly once with only the media. So if my pH is as per my satisfaction, I'm going to be adjusting my CO2 accordingly. So for 7.33, the CO2 on the display might sometimes be 6.2 or 6.3. It doesn't matter really. For me, the pH is more important than the CO2 display on the incubator.
More to do with the calibration of the incubator as such.
Yeah.
Any more questions? I think there is no. Thank you very much, Shubhangi, for the very basic and very important thing what you explained thoroughly. Akash, to take over.
Yeah. Thank you so much, Dr. Shubhangi and Dr. Shrikant Yatnale. Now we'll be moving on to the last part of our session planned for today. And we have saved some of the best for the last. And taking us forward would be Dr. Sapna Srinivas. She's the lab director at Mamta Fertility, Hyderabad. And her CV is only one line because she feels this is the most important thing for her. She is been an invited guest as lecturer for at multiple national conferences, presented papers and all this, but she feels this is the best thing for her to put her on CV. So it's all over to you, Dr. Sapna.
Thank you, Akash. It's an absolute pleasure to be here today, and good evening, everyone. A very good morning to Dr. Joe Conagan, who's there with us today from San Francisco. It's really pleasure. It's my pleasure to introduce you to him. Dr. Joe, who is currently the lab director of the Pacific Fertility Center in San Francisco since 1999, and also a teacher at the UCSF IVF program, actually grew up in a small fishing village called Killybegs in Ireland, and obviously was fascinated in marine biology. He started his embryology career in London's Hammersmith Hospital, and this was where he laid his foundations in embryology in the early days of IVF. He graduated with a PhD in embryology from the University of London and then moved to Pacific Fertility Center as a partner in San Francisco, a city that he claims he loves and lives with his wife and 3 children. He's a board-certified high-complexity clinical laboratory director, and over the next 30 minutes, I invite you, Doctor, to take us through the dynamics of culture system. He's a strong advocate of single embryo transfer and therefore believes in high standards for culturing and vitrifying embryos. We are honored to have you with us, sir, today, and the floor is all yours. I request the delegates to bombard us with questions because we have none other than Dr. Joe Conaghan, and Akash has already mentioned that we've saved this, the best for the last. So please bombard us with questions and let's make use of this beautiful personality who's on board today. Over to you, Dr. Joe.
Okay. Thank you, Dr. Srinivas, for that very kind introduction. Can you hear me okay?
Yes, we can.
Okay, let me see if I can share my screen with you right here. All right, that looks good from my end. Can you see this okay?
Yes, Professor, we are good. We can see.
Okay, perfect. Thank you. Well, thank you very much for the invitation to be here today. It's a great pleasure for me to participate in your meeting. It is very early morning here in San Francisco. It is 6 a.m. here.
And I saw you on the screen at 5 a.m. too. And it's absolutely an honor for us to have you with us.
Yes, and
thank you so much.
I'm very glad that I got to listen in to the previous speaker and also to the previous session because I think they have also some relevance for what I'm going to say. We are running a little late today, I think, so I will try to be as brief as possible so that we can get some time for questions at the end.
Right.
So I'm going to talk to you about embryo culture, and I think what I'm going to say is going to complement what we've heard already from the previous speakers. And we just heard a very nice talk about preparing dishes for IVF cases, and I'm going to get into a little more detail now about the specific components of the culture system. And we can break down the culture system into many parts, the lab itself, the people who are doing the work in the lab, the actual culture media that we use. And we don't have time to talk about all of these today, so I'm going to go through the important things that I think are relevant for doing good embryo culture in the lab. And I'd like to start by just pointing out very briefly, the previous speaker talked about dish preparation. And if you can see here in this little picture right here, here is a culture dish with some micro droplets of culture medium under oil. And this is a common culture system that all of us use in our labs today. And it's actually a very old culture system. It was developed by this guy, Ralph Brinster, back in 1963. It's even older than I am. So sometimes we think that we have very modern and very sophisticated technology in our labs, but in actual fact, what we are doing has been around for a very long time. And we know when something has been around for a long time that it's probably very good and works very well. So still today, we have yet to come up with a system that is better than this one that was established over 50 years ago. So the plan for my talk today is to talk specifically about some of the elements of the actual culture system. And we've heard about some of these already today. We've heard a little bit about CO2 and pH in the last talk, and I'm going to go into that in a little more detail. I'm going to talk about creating a low oxygen environment, as was discussed in the previous session, and which I think is quite important. And then in the second half of my talk, I'm just going to talk a little bit about our experience with culturing embryos to day 7. We do this routinely for every patient. We culture all embryos to day 7, and we have a single-step culture system, which I think is going to be of interest to our listeners this morning. So for most of us in the IVF industry, we are using a gas phase in our lab that always has CO2. Sometimes people are using carbon dioxide in air, and sometimes people are using carbon dioxide in combination with a low oxygen environment. And these are the typical systems that are used in IVF labs around the world. Some of us use 5% CO2, some of us use 6% CO2. Again, the actual CO2 concentration, I would just like to point out what the previous speaker, Dr. Gangal, said. The actual concentration of CO2 is not that important. It's the pH of the culture medium that's important. And as embryologists, we should be measuring the pH, and we should be adjusting the CO2 concentration in the incubator to get to the desired pH. So our previous speaker likes a pH of 7.33, which I think is excellent. And whatever CO2 concentration you have to use to get to that pH, then that's what you should go with. And you shouldn't be a slave to trying to get exactly 5% or 6%. pH is what's important. Now, in the previous session, they talked about low oxygen culture. And oxygen is a very aggressive molecule at the cellular level. And no cell in the body sees 20% or 21% oxygen, which is what we have in the atmosphere. So it makes a lot of practical sense to culture in a reduced oxygen environment. But also there are many publications that show that the low oxygen environment gives us better development, higher cell numbers in our embryos, and better outcomes. And I think that we have to consider that culturing in CO2 and air that's a high oxygen environment, and it's probably quite stressful for embryos. So if we're doing short-term culture, if we're doing transfers on day 2 or day 3, maybe it's not going to have so much of an effect. But in my lab, for example, we culture embryos all the way to day 7. So I think culturing in a high oxygen environment for that length of time is probably very stressful for embryos. So we always go for a low oxygen environment. Now, I still have many big box incubators in my lab, and these large incubators have a incubation chamber that is in my lab 150 liters in size. Right? Some of you may have smaller chambers that is maybe 100 liters, but in my lab we have chambers that are 150 liters. And when we want to create a low oxygen environment in this large chamber, we have to put a lot of nitrogen in there to depress the oxygen concentration. So in this big incubator, every time we open the door and close it, we have to put in about 135 liters of nitrogen gas in order to depress the oxygen concentration again. So we're using a lot of nitrogen gas. And historically, and what I see when I travel around the world, is that a lot of labs use these cylinders of compressed nitrogen gas to create their low oxygen environment. And this is actually not a very efficient system, because you have a very limited amount of nitrogen gas in these cylinders, and you have to change these cylinders frequently. And if you're using big box incubators, you're probably changing these cylinders every day. Now, there is a better way. And what we use here in San Francisco is we use liquid nitrogen to generate the gas, to generate the nitrogen gas that we use in our incubators. And this is a very simple thing to do. You're all familiar with using dewars of liquid nitrogen. And the nitrogen dewars that come in from the supplier, as you know, they have an outlet where you can draw off liquid for other purposes in the lab for your vitrification needs. But if you look at the top of the cylinder, you have many other options right here. And one of the options that you have, so here is your liquid outlet right here. And another option that you have is that these cylinders also have a gas outlet, and you can draw the gas off of these cylinders to use as the nitrogen gas in your incubators. Now, the one thing that you have to remember is that if you're going to do this, if you're going to use liquid nitrogen to generate gas for your incubators, you have to use these high pressure tanks, tanks that are at 230 PSI. Pounds per square inch, because if you use a normal liquid nitrogen cylinder, you won't get enough pressure in your gas to keep your incubators happy. And for those of you who are using the compressed gas cylinders, the attachment where you attach your hose right here to get your gas, it's the exact same hose and the exact same attachment that goes on the liquid nitrogen cylinder. So whatever hose that you have or whatever connection you have, if it's a regulator or if it's a direct hose right here that goes to your compressed gas, you can take that same connection and you can connect it up to the gas outlet on your liquid nitrogen dewar. So it's the exact same hardware. It's no extra expense. And it's a lot cheaper to use this method for generating nitrogen gas than it is to use the compressed cylinders, because compressed cylinders you're changing all the time. These guys you don't have to change very much. You can also use a regulator right here as an option to get the gas out of the liquid nitrogen dewar. And if you use these guys, you have to change them every day. If you use these guys, you only have to change them about every 2 to 4 weeks, right? So a large cylinder of compressed gas will give you enough nitrogen for these large incubators right here to fill your incubator about 40 times. Every time you open the door, you lose your nitrogen and you have to refill it, so you can get about 40 door openings. Whereas if you go to the large cylinder of liquid nitrogen, you will have enough nitrogen to fill your incubator about 1,500 times. So it's a much more efficient system. And again, when I travel around the world, I'm surprised that more people don't use this system, because this is a much more efficient way to supply your incubators with nitrogen and to create that low oxygen environment. So it's much more cost effective. And also the nitrogen that you're getting, because it's evaporated liquid nitrogen, it's distilled nitrogen. So the gas is extremely clean. It's probably the cleanest nitrogen gas that you can get. So it's very efficient and it's very clean. And I think it's a system that we should all consider using. Now, let's move on and talk about carbon dioxide in the incubator. And we heard a little bit about this from the previous speaker. It's morning time here in San Francisco, so I'm having my coffee. I'm having my morning coffee. And for those of you who drink coffee, you will know that during the coffee brewing process, when you pour your hot water on the coffee grounds, you see all of these bubbles forming, and this is carbon dioxide being released from the coffee grounds during the brewing process. Now, when you make your coffee in the morning, if you don't do it correctly and you don't allow the carbon dioxide to be released here, then your coffee is bitter, because carbon dioxide is an acidic gas, right? So when you make your coffee, you want to get the carbon dioxide out of the coffee so that your coffee is not going to have a bitter taste, right? Now, in the IVF lab, we have the opposite situation. We want to get the carbon dioxide into our culture media, right? We don't want to get it out like we do with the coffee. We want to get it into the culture media, right? Now, as we heard in the previous talk, it's very important that we prepare our dishes correctly and that they're fully equilibrated in the CO2 environment before we use them, because we want the pH to be in the physiological range before we ever put an egg or an embryo into that dish. In the old days, we used to have phenol red in our culture media, so we could see if the media was at the correct pH or not. But today, many media do not contain phenol red, so we do not have this visual signal telling us whether the media is at the correct pH or not. And for those older embryologists on the call, you can see how pink this media is right here. And the pH is not quite correct because this media is a little too pink. The pH will be too high, and we have to fully equilibrate this dish before it is ready for use. Now, when we think about equilibrating dishes and we think about pH in the lab, it's worth asking ourselves a few questions, right? So one question that often comes up among embryologists is, if I have a pre-equilibrated dish that's been in my incubator for 24 hours and I take it out of the incubator, how much time do I have to look at the embryos or to look at the eggs in that dish before the pH starts to change, right? So that's one question. Another question that sometimes comes up is, if I forgot to make up a dish for a patient, how long does it take for a dish to equilibrate? Like if I forgot to make up a dish, can I make up a dish today and it will be ready in maybe 2 hours or 4 hours or 6 hours, right? So that's something that we also can ask ourselves sometimes. And then last question, can I do ICSI in a dish of culture medium, right? So these are sort of common things that young embryologists should know and understand and should be able to answer. And the first question, when you have a fully equilibrated dish that's been in the incubator, let's say for 24 hours, right? And you've reached a pH of maybe 7.33, just like our previous speaker was recommending, right? And we take that dish out of the incubator, how long do we have before the pH changes? Well, we've measured this in our lab and we made up a dish and we took it out of the incubator for 1 minute. We took it out of the incubator for 5 minutes. We took it out of the incubator for 10 minutes. And we could not measure any change in pH until we took the dish out of the incubator for 10 minutes. And that's the graph that you're seeing here. So the dish was pre-equilibrated here to about 7.33, 7.34. And then this little flat area on the graph shows where we took it out of the incubator. And even after we put it back into the incubator, the pH was still the same. But then there was a sort of delayed reaction whereby the pH went up a little bit, but not significantly. It only went up as far as 7.37. So arguably it's maybe still in the physiological range. And then over the next sort of 3 or 4 hours, the pH came back down to where the starting pH was. Now, we were only able to measure this difference here when we took the dish out of the incubator for a full 10 minutes. When we took the dish out of the incubator for less than 10 minutes, we did not see any change in pH, right? So when you, as an embryologist, are working with a dish of eggs or embryos, you have up to 10 minutes to do what you have to do with that dish, to score the embryos, to move the embryos, to clean off the cumulus cells, whatever it is you need to do. You have 10 minutes of working time before the pH will measurably change. So everything that you do in your lab should be designed to have dishes of culture medium out of the incubator for 10 minutes or less. If you have them out of the incubator for 10 minutes or more, the pH will change, it will go up, and it may go out of the physiological range. Now, what happens if you don't have an oil overlay? Well, if you don't have an oil overlay, the dish will equilibrate very quickly. It will come down to the pH that you want very quickly, usually in less than an hour. But as soon as you take that dish out of the incubator, the pH starts to rise. You see right here, the pH goes up immediately. This dish right here was only taken out of the incubator for 1 minute, and you can see that even during that 1 minute excursion from the incubator, the pH went up to like 7.55, right? So if you don't have oil over the media, the pH will change faster than you can believe, right? And some of us use culture media for doing embryo transfers or for maybe doing other procedures in the lab where we do not have oil over the media. And I do not recommend this, because if you have any dish where you do not have oil over the culture media, the pH immediately starts to change when you come out of the incubator, and it goes outside of the physiological range, and it creates stress for your embryos. So really, you should never have any culture media in your lab in a dish where you do not have an oil overlay, because the pH is going to be out of your control. Right. Now, what about making up a dish? Let's say you forgot to make a dish for a patient and you need to make a dish today. Can you make a dish of culture medium and use it the same day? The answer is no, right? Now, we've measured this again, and again, this comes back to the previous speaker. When you take some oil and some culture medium out of your refrigerator and you make up a petri dish with culture medium under oil, the first thing that happens is that the pH of the medium actually starts to rise, because as the dish warms up, CO2 comes out of solution. But then as the CO2 in the incubator starts to dissolve into your media, then the pH of the media starts to come down, and it takes a long time to come down. You can see how many hours right here. So if we want to get to a pH of about 7.34, we need absolute minimum of maybe about 8 hours to get close to that pH. And really we need something like 12 hours for a dish to fully equilibrate, right? So when you're making your dishes, and the previous speaker told us that she makes her dishes the afternoon before, and this is important, your dishes need a long time to fully equilibrate, because it takes a long time for the CO2 to go through the oil and get into the culture medium. So if you're making up dishes in your lab of culture medium, please allow at least 12 hours or a minimum of 8 hours for the dish to equilibrate before you try to use it with eggs or embryos, right? So here are the answers to our questions, right? If we take a pre-equilibrated dish out of the incubator, how much time do you have? You have 10 minutes or less, right? You always need to be working on your dishes for 10 minutes or less, and then you need to get them back into the incubator. If you have no oil over the dish, you have no time. The pH will start to change immediately. If you're in a situation where you forgot to make a dish and you need to make one, you need at least 8 hours for that dish to fully equilibrate, and really you need 12, right? And then finally, if you're doing some procedure like ICSI and you think that using culture medium is a good idea, and I've seen this in different labs, it is not a good idea, because you do not have time to do these micromanipulation procedures in the dish of culture medium before the pH will start to change. Now, last thing I want to say is just very quickly about temperature. We all know what the importance of temperature is. What I have noticed in our lab is that we do have some places in the lab where we have these stainless steel surfaces So this is a microscope stage right here with a heated stainless steel surface. And in some places in our lab, the stage is not heated. For example, in the area of the lab where we do vitrification, we do not have a heated stage. It's a room temperature stage. And this is a metal stage, and metal conducts heat very well. So if you take a dish of oocytes or if you take a dish of embryos and you put it on a metal microscope stage that is not heated, that will suck the heat right out of your dish, because metal conducts heat very, very quickly, very well. And you've got to be very careful. Like if you're doing vitrification of maybe a couple of embryos from this dish or some eggs from this dish, you've got to be careful about having the dish on a cold metal surface, because the dish will cool down incredibly quickly. All right, now we heard in the previous session a lot about blastocyst culture, and I wanted to sort of finish off today just by talking about our experience with blastocyst culture and about some of the things that we've done to really generate more embryos for patients. And specifically, I'd like to talk about our experience with culturing embryos to day 7 of development. This is the system that we use in our lab. So we use these Petri dishes with drops of culture medium around the outside and some wash drops in the middle right here. Our drops for embryo culture are 50 microliters, right? So these are 50 microliter drops, and we put 5 embryos in each drop. And we put them in there on day 1. So after we do our fertilization check, we put 5 fertilized eggs or zygotes in here, and we leave them here from day 1 to day 7. We do not change the dish, we do not change the media, and we do not check them on day 3. We leave them undisturbed. So after day 1, the first time that the embryos will get checked will be day 5, and then also on day 6 and also on day 7. But between day 1 and day 5, it's undisturbed culture. We do not take the dishes out of the incubator. Now, we like the group culture system. Like I said, we have 5 embryos per droplet, and as we've heard previously, we get the autocrine effects of the growth factors that are produced by all of these embryos growing together in the little drop. We do have to worry about nutrient depletion from the drop and waste buildup, but I think overall the benefits of group culture are better than the benefits of single embryo culture, where you have this lonely little embryo growing all by itself. You know, there's going to be less waste and more nutrients available to this embryo, but you're losing this synergistic effect that you get from group culture. So we first got interested in day 7 embryo culture back in 2013 when this paper was published in Fertility and Sterility. And we hadn't really thought about day 7 culture before this, but in this paper they showed the blue bars right here are their day 5 embryos, and this is implantation, pregnancy, and ongoing pregnancy rates. And you can see that the day 5 embryos are the best. And then the yellow is the day 6 embryos. You can see that the day 6 embryos are not as good, but still not too bad. And then the green bars right here show their experience with day 7 embryos. And you can see that with day 7 embryos, they had about a 30% implantation rate, 37% ongoing pregnancy rate, and a 37% clinical pregnancy rate and a 37% ongoing pregnancy rate. So we were very interested to see these numbers. So we, the following year, 2014, we started culturing all of our embryos to day 7. And what we decided was that because the results are not so good on day 7, we're going to be very strict. And in order to use a day 7 blastocyst, we must see that the embryo has continuously developed all the way to day 7, so it must not have arrested. And we also will only vitrify good morphology blastocysts on day 7. So if the embryo is not very nice, we're not going to vitrify it, right? So here are some examples of some embryos that we've seen on day 7. And if we ask the question, would you freeze these embryos on day 7? The answer for this one is no. Yes, this one we would freeze because I think this one has more trophectoderm cells. It has better morphology. It has clearly got a nice inner cell mass right here. So this one we would freeze, and these other guys we would not freeze. So we set the bar very high on day 7. We're not going to freeze just any embryo. We want only good quality embryos on day 7. Now, since 2014, we have biopsied a lot of embryos. We have biopsied almost 20,000 embryos in our lab in this time period. And you can see of all of the embryos that we've biopsied, about a quarter of them are day 5 embryos. Most biopsies we do on day 6 because we like the embryos to have more cells when we do the biopsy. But look at this, over 10% of all of the embryos that we've biopsied have been done on day 7. So day 7 does contribute some extra embryos, maybe about 10% more than if you stop doing embryo culture on day 6. Now, because we're doing biopsies, we have the luxury of looking to see, okay, are the day 7 embryos any different than the day 5 or day 6 embryos? Well, here is the average euploidy rate. So normal chromosomes in embryos under age 35, we expect that about 50% of embryos will have normal chromosomes. In egg donor, oocyte donor patients, we expect about 70% of embryos to have normal chromosomes if they're day 5 or day 6 embryos. And you can see the numbers come down as the patients get older. Now, if we add the day 7 embryos in here, you will see, so the blue bars are just the same numbers that I showed you, but you can see in every age group, the red bars right here, the day 7 embryos, the number that are euploid that have normal chromosomes is less in every group. So day 7 embryos are not the same as day 5 or day 6 embryos. They have a lower rate of normal chromosomes. Now, here are our implantation rates for day 5, day 6, and day 7 euploid embryos. These are euploid embryos, normal chromosomes, okay? You can see that day 5, which is shown in blue right here, and these are all the different ages starting from less than 30 all the way up to 42. And you can see that the implantation rate, when we transfer a single euploid day 5 embryo, the implantation rate is in the sort of range of 70%, right? And over here on this side, you have the overall implantation rate average for all groups is 67% with a day 5 euploid embryo. Now, for day 6 embryos, which are shown in red, you can see that the, you can see that the implantation rate is maybe about 10% lower. So it's almost as good, but not quite, right? So day 5 and day 6 embryos perform very well, but look down here, day 7 embryos, not so good, right? So the implantation rates, these are day 7 euploid embryos, normal chromosomes. You can see that the implantation rates really are averaging somewhere in the sort of 20 to 25% range. And the overall implantation rate actually averages out at 23%. So you can see that's a lot different than day 5 and day 6 embryos. So the question is it worth doing day 7, right? And I think for a lot of you on the call today, you probably end your embryo culture on day 6. Well, here's our experience with day 7. We've been doing it now for a long time. So we now have over 4,000 patients where we cultured them one more day. And of all of those patients, about 30% of those patients got one or more embryos that we vitrified on day 7. So definitely there's a benefit to the patients, right? The number of patients who only had day 7 vitrification, right? They had nothing frozen on day 5 or day 6, only day 7 is very small, just 3% of patients, just over 100 patients. We had over 2,000 more embryos vitrified over this period of time. And out of those 2,000, you can see that only about 1/3 had normal chromosomes. So in your day 7 embryos, only about 1/3 of them are going to have normal chromosomes. So this is not very efficient. With day 5 and day 6 embryos, we average about 50 to 60% with normal chromosomes. Day 7, it's only 1/3. And then we had all of this extra freezing that we had to do because we were going to day 7. So there's a lot more work for the lab. And then the other problem that we have with day 7 embryos is that we don't get to use them very often. We don't get to thaw them very often because patients will use their day 5 embryos first. If the day 5 embryos don't work, they will use their day 6 embryos. And if the day 6 embryos don't work, they will use their day 7 embryos. So it's only patients who either have no day 5 or day 6 embryos or have used them already that will come back for a day 7 FET. So in all this time, we've only done 200 FETs with day 7 embryos, even though we've been doing this for a long time now. And from those 200, we have 44 children born. So is it worth it? You can see the efficiency here is not very good. This is a lot of extra work for the lab. But if you ask these patients that have these 44 children, these patients will tell you, yes, day 7 embryo culture is worth it, right? And our data is very similar to other data that's been published in the literature on day 7 embryos. So this is just an example of one paper that was published in 2018. And you can see with day 5 embryos, this group have a live birth rate of close to 50%. But with day 7 embryos, their live birth rate is 25%, similar to what I showed you on our data. So you have to ask yourself the question before you launch into this: is it really worth culturing all embryos to day 7 for the small number of babies that you're going to get from this process? Because it's going to be a lot of extra work for your lab. All right, so I'm at the end, and I'll just summarize what we talked about today. So I talked about the use of liquid nitrogen to generate nitrogen gas to create a low oxygen environment in your lab. This is a very efficient system. It's very cost effective, and it saves you a lot of time because you're not changing these tanks of compressed gas. And it's a very clean gas because it's distilled nitrogen. We talked about the importance of CO2 and pH in the lab, and maybe this is the most important thing that we talked about today. And then I finished up by talking to you about our experience with culture to day 7. We use a very simplified culture system. We make up the dishes 24 hours in advance. We put in the fertilized eggs or oocytes into a single continuous culture system. We do not change the media. We do not change the dish. We leave them undisturbed up until day 7. And then we choose currently to end our culture on day 7. Many of you, I think, ended on day 6. But day 7 is also a possibility if you want to create a few more embryos. And I think that I'm a little bit over time, so I'm going to stop there, and I'm happy to take any questions. Thank you, everybody, for being here today and for listening to my talk.
Thank you so much, Professor. That was absolutely wonderful. So crystal clear, so simple, so typical of somebody who's been doing wonderful work for the last 25 years. So I would like to start by asking some questions that we have here. The first one by Ekta. If we freeze on day 6, the embryos, so do we transfer these embryos on day 4 or day 6 when they are thawed? What are your, how is it, what is your protocol? What do you follow in your lab during the frozen embryo transfer cycle?
That's a nice question. So we do frozen embryo transfers on day 6 of progesterone. So when patients
are on day 6 of progesterone is day 5 of the endometrium.
Yes.
Because you started on day 0.
Yes.
Right. So day 5, so a day 6 embryo goes into a day 5 endometrium.
Yes, any embryo. So when we prepare the patient for a frozen embryo transfer, we do not take into consideration if it's a day 5 or a day 6 or a day 7 embryo. We always transfer the embryo on day 6 of progesterone or the day 5 endometrium. So even if it's a day 7 embryo, we do the same thing.
Okay. The next question, which component of culture media is responsible for pregnancy loss? Is it possible to have good embryonic development, but pregnancy loss at the same time? What can be the reason leading to this, and how can we improve it? This is from Dr. Sarabjit Singh.
So I love that question. I have seen, I've been doing this for a long time, as many of us have on the call, and I've seen a lot of labs where they have a very high rate of biochemical pregnancies or even a high rate of miscarriage. And we know from the literature that the number of euploid embryos that we see in the lab changes from one lab to another lab. Like if you look at some of the publications from some of the companies that do the genetic analysis for our embryos, like Reprogenetics, for example, has been a good example in publishing data. They have shown that in some labs, you see much higher rates of normal embryos than in other labs.
Great.
And this is kind of strange, right? But also as embryologists, we know that sometimes we see a higher rate of biochemical pregnancies than we're used to, or a higher rate of pregnancy
Professor,
loss.
can I stop you and ask you one question here? Do you think it is because of the meiotic errors or the mitotic errors that are happening due to the lab conditions, the mitotic, or is it because of the preparation of the patient, the meiotic? What, in your view, is the key driving force in this scenario?
I think the meiotic errors we have no control over,
right,
because we don't have control of stimulation, we don't have control of the patient, we don't have control of the biology of the eggs that we receive. So this we don't have control over, and this will be usually very closely related to the patient's age.
Right.
But the mitotic errors, I think, when these occur, at least in some part, can be because of poor embryo culture conditions.
Right.
And I think that the reason that we sometimes see in some labs high rates of biochemical pregnancies or high rates of pregnancy loss or higher than expected rates of aneuploidy is probably because the culture environment is very stressful.
Yes.
Right? Now we talked today, myself and Dr. Gangal talked today about embryo culture and about things in the culture environment that can cause stress. For example, we heard about osmolality in the previous talk, right? Maybe the osmolality is not quite right in the culture environment. Maybe the oxygen concentration is high. Maybe the culture conditions are not perfect and the embryo is stressed. So therefore there are mitotic errors that are occurring during embryo culture. And I think when we see lots of biochemical pregnancies or lots of pregnancy loss, we have to take some ownership and we have to look at our culture system and see maybe the culture system is not as good as we think. Yes, we are getting some blastocysts, but maybe the blastocysts are not healthy because we have created a stressful environment.
Right, they're highly mosaic. Professor, we lost you?
Yeah. No, I'm here.
Oh, you're there. Okay.
Yeah.
So you would, how would you, if extending Dr. Sarabjit's question, how would you troubleshoot? Where would you start? Supposing we have a little drift and we're seeing a little bit of biochemical pregnancies, suddenly a lab that's doing very well, the KPIs are going okay, fertilization rates are okay, cleavage is okay, but then we're starting to see this. Where would you start? How would you guide us? Would you troubleshoot in this scenario?
So the first thing that happens in this situation, I think, is always a mistake. The lab thinks they should change the culture media.
Right.
Like that's a very common reaction when things are not going very well, because you have a friend who's an embryologist in another lab and they're telling you about the great blastocyst rates that they have and the great pregnancy rates that they have.
Right.
And you're looking at your own data and you're thinking, my data are not so good.
Right.
And you think, I'm going to change the culture media because they have a different culture media than me. And if I change my culture media, I will have the same outcomes as them. That will not happen.
Right.
That will not happen. Now, I think that the culture media quality that we have in IVF today is very, very high. So it doesn't really matter which company you buy your culture media from. All of those companies, they make a very good product. If they made a bad quality product, we wouldn't buy it. Right?
Yes.
So do not change the culture media. The culture media typically will not be your problem. That, as long as the QC on the culture media is okay, then you don't need to worry about the culture media. If you're having problems, it means that you're doing something wrong with the culture media. Maybe you're not making your dishes correctly, right? Maybe you're making small drops in a laminar flow hood and the media is getting to evaporate a little bit before you put the oil over the top. Maybe the pH of the media is not exactly correct. Now, in our lab, we heard from the previous speaker that she tests the pH of the media once a week.
Right.
We test our pH every day.
That's what I do too. We test
Every single day.
Yes.
In every incubator. And what we do is we make up the media exactly as we are going to culture embryos. So we add the protein and we put that media in the incubator, and then we test the pH the following day after equilibration. And we do that every single day. Now, that's probably too much. We probably don't need to do that much. Like testing it once a week is fine. The important thing is that you are checking it. You must be checking the pH, right? Now, what I would recommend, just to answer your question more directly, is that if you have a culture system and your embryos are not growing very well, or maybe your embryos are okay, but your pregnancy rate is not very well, the best thing that you can do is find a friend, find another embryologist who's using the same culture system as you, and do a side-by-side comparison to see what am I doing and what are they doing, right? What is the pH of their medium? What is the osmolality of their medium? Do they change the dishes on day 3? How many embryos are they culturing together? Try and benchmark against someone that you know is doing very well, right?
What about the air shift quality? Anything transient in the air that could go wrong?
Air quality is very important. I'm in California right now, and we have a lot of fires burning in California, so we have a lot of smoke in the air. Yes. So we
worried how you're managing it.
Yeah, we are very sensitive to the air quality. So one of the things that you can do for your air quality is you can keep your embryos in the incubator, right? Because if you fill your incubator with distilled nitrogen gas, carbon dioxide, that is like almost the entire environment inside the incubator. So there's no outside air going in there. There's very little or no outside air going in. So
that makes sense. Your uninterrupted culture makes sense because you're not going to open the door very often.
Yes, and a lot of embryologists.
Why is it that you're using the box type incubators? When so many people are talking about the bench tops, how come? I'm really curious about that.
We are transitioning from the big boxes to the small incubators, and you know, we don't like to throw away equipment that's working fine. So every time a big box incubator is dying or it's not working anymore, then we will get rid of it. So maybe in a couple of more years, we will not have so many big box incubators. But right now we have a combination of both.
So we'll move to the next question.
Thank you for all the great questions, by the way.
We are honored to have you, sir. So preparing the nitrogen from liquid nitrogen, do we need to worry about the purity of the gas? Do we need to use any filters? When purchasing compressed cylinders, how to check for the purity of the gas apart from the purity certificate that the seller is providing?
Yes, that's very difficult. And the problem is that those cylinders get very dirty. They move around to lots of different industrial applications. And even if you check the purity of the gas, if you go to all the trouble of trying to check the purity of the gas in one cylinder, it will be different in the next cylinder.
Okay.
So it's very hard to check that. So that's another reason why, if you can avoid it, you should not use this gas for your incubators. If you can, you should use liquid nitrogen evaporation because it's very efficient. And like I said, it's distilled gas. So it's the cleanest gas that you can get. Now, the other thing that you can do is you can put some filters in between the gas supply and the incubator itself.
Like the online coda?
Like the online coda, right? It's a carbon filter and it will take out a lot of impurities out of the gas. So that's also an option. But remember, in your incubator, like 90% of the environment inside the incubator is nitrogen gas.
Right.
So you really need to do everything that you can to make sure that that's the cleanest gas that you can get.
The next question, sir, is what method do you use to check the contamination level of liquid nitrogen? I think you've already answered that.
Ma'am, you can see the further question in chat box.
Yes. In chat box? Okay,
Yeah,
I'll move there.
yeah.
Do you advise all day 7 embryos to get PGS done before they go for transfer?
No, not at all. I think we're doing too much PGS or PGTA. We do it too much here. So no. But what you got to remember is that if you take if you take 100 day 7 embryos, only about 30% of them will be genetically normal, right? So from your 100 embryos, only 30 are normal. Now from those 30 normal embryos, you're only going to get about 25% are going to give you a baby, right? So out of 30, maybe you're going to get 7 or 8 babies, right? So it's not very efficient. So if you think about, you started with 100 embryos, you end up with maybe 7 babies. So the efficiency is only going to be about 7% live birth rate, definitely less than 10% live birth rate if you don't do PGS. So I just presented the data today. I don't want to make any decision for anybody. It's not very efficient. You can see over the years how many embryos we've biopsied, how many extra freezings that we've had to do. It is definitely not very efficient. So each embryologist has to decide for themselves, is it really worth it? Is it worth all this time just for this small extra number of babies?
Yeah, but we get 1 embryo extra for that patient, and therefore there's no harm in culturing, leaving the embryo for 1 more day instead of discarding on day 6. That's what you told us. That's a very important point. And if there is that 10% of patients who had that 1 extra embryo that goes into the freeze, and if it's her only embryo, then you're giving her a chance. So that's our take home from today.
you're giving her a chance, and you know, if you don't do genetic testing on the embryos, then you just tell the patient, okay, we have a day 7 embryo.
Yes.
The chance maybe at best is 10% that you will get a baby,
but at least you have
probably patient will take that choice. She will be happy.
Absolutely, instead of discarding that. Professor, next question. Is there any difference in the blast developed in a CO2 incubator and a trigas incubator, even with good blast stimulation rate and good morphology?
We have both types of incubators. We have tri-gas incubators, and we also have the incubators that mix their own gas. And we do not see any difference in the quality of the embryos, really, that, you know, we can't measure any difference in quality. I would just caution you to make sure that with the tri-gas that you also measure the pH of the media on a regular basis, just to confirm.
But you can't control the CO2, so you just get what you get in the tri-gas.
You get what you get, but you got to always be checking the pH to make sure that your manufacturer is definitely giving you what you're asking for. And, you know, again, it's just taking a little bit of time to do these measurements just to make sure that everything in your lab is running nice and tight.
Do we have time? Dr. Ved, are you there? Can we continue? Professor, are you okay? Can we take one more question?
I can take one more question, yes.
Okay.
You can take one more question.
Okay,
there is
Dr.
no problem.
Okay, Dr. Ved. Can we keep media for equilibration in test tubes with volume of 15 ml, or do we have to prepare the dish and then equilibrate? That's a good one.
So if you keep media in a test tube, 15 ml test tube, you got it. So let's think about this, right? The test tube is like tall and narrow, right? And the surface area of the media is going to be very small relative to the volume. So it's going to take a long time to equilibrate.
Yes.
I don't know how long, because I haven't done that specific measurement, but we saw with the microdrops how it takes 12 hours to equilibrate. So with a test tube of media, probably it's going to take 24 hours or more to equilibrate, just because the surface area where the gas is able to diffuse in is very small relative to the volume. So yes, you can keep test tube of media equilibrated in the incubator like that, but just be aware that it's going to take a long time to equilibrate.
Does that increase any ammonia buildup, or will that add on to the ammonia buildup that we're going to have because of this long extended culture? Because
potentially.
We're going to take longer time.
Yes,
So
yes,
it's always better to do the micro droplets and leave them inside. You keep your dishes open, Professor? You keep the lids open in the incubator?
No, in that slide that I showed you, we were just looking to see, you know, this was an experiment that we did where we had media in a dish, and we had no oil.
Yes.
So we took it out of the incubator, and the pH went up immediately. And then we put it back into the incubator, and we found that when you put it into the incubator with the lid on the dish, that is the slowest way for the gas to get back into the media.
Right.
Like if you can tilt the lid of the dish a little bit to let the gas in, or take the lid off, then the gas will come back.
We keep it at 45. We just keep it at 45.
Yes. So
that
will definitely speed up the equilibration of the media if you keep it at 45.
Absolutely a pleasure. It was just an honor, and we are so delighted. Namaste with all our love from all of us here. There are 140 people waiting for you. They've been here from 2:30 in the afternoon, and we are honored, Professor. Thank you so much for being here with us. And over to you. Thank you so much. It was absolutely
I hope I can come
wonderful.
and visit you in India again soon.
Yes, we would love to have you. We would just love to have you.
Yes, as soon as possible.
As soon as possible. Professor, one last question. What are you doing differently in the COVID times?
You know, we have divided the lab staff into two teams. So we have team A and team B, and each day only one team is working.
Okay.
And that way, because if one team get COVID,
right,
then the other team is clean. Yeah. So from the people that I work with in the lab,
yes,
50% I have not seen them since March.
Oh my God.
Okay. Okay.
Yeah. So that's kind of sad, but that's how we're working.
Yes. Thank you so much. Over to you, Dr. Ved. Thank you so much for having all of us here.
Thank you. Thank you, Dr.
very
Sarna,
much.
and thank you, Dr. Konagan. Is Dr. Charudat, you are here?
Yeah, he is there, but unmute Charu.
You're on mute, sir, Charu, sir.
That's
Hello.
a wonderful picture.
Yeah, he's
Is that how you
Yeah,
look now in
am
the
I
lab?
Hello.
Yes, Charu, please. Please go ahead for vote of thanks.
Yes. So, okay. So, on behalf of IRA, I would like to thank all of our distinguished faculties, particularly the international faculties, Dr. Joe, sharing their knowledge with us, and we are all delighted to have all of you and enlightened with your experience. Similarly, I would like to thank all my national faculties, moderators, my dear friends who have contributed a lot and take all the pains to be here all the day and conducting and moderating the session. And not but the least, I would like to thank all our team members who have taken a lot of efforts from last so many days to make this program, finalize this event, and get it done in fantastic way. And to begin with, this today's start is really fantastic. I hope everyone has enjoyed. And I would also like to thank all my participants who have participated, who have attended this workshop from since afternoon, and they are still there. And we hope to have a fantastic session as tomorrow also. So I hope to see all of you again tomorrow for the wonderful session coming up. So once again, thank you all on behalf of iHera to be part of this event. Over to you, Dr. Ved.
Thank you. Thank you, Dr. Charu, and thank you all of you and my national and international faculty, and of course, Cryo Biosystems for supporting this event. And I would like to invite you all for tomorrow mega event. It will be from 9:30 to 7:30 onwards tomorrow. So please join us for tomorrow. And thank you so much once again. Thank you. Thank you. Dr. Shukla, are you there? You want to say something?
No, I'm overwhelmed. So I do not have words to say. But yes, it was great watching all those participants waiting for almost 3 hours, more than 3 hours. And not just waiting and listening, but also interacting very regularly and very enthusiastically. So it is, yes, very morale boosting that so many participants are there. And hope to see all of those again tomorrow and maybe many more. Have a wonderful evening. Thank you.
Thank you. Thank you so much. Thank you. Thank you. Thank you.
Sir, shall I end the session?
Yeah, you can. Please go ahead.
Okay, thank you very much. Thank you very much.
Thank you, everybody.
Thank you.
Thank you.
Thank you. Bless you.
Thank you all. Thank you all.
Thank you.